मैंने ही ऑफ किया था एलेक्स। हाय सर्वजीता। हाय राशि। हाय गिरीश। एम्स का फॉर्म आ गया पैरामेडिकल वाला, मुझे भी पढ़ना है पैरामेडिकल। सच्ची में आ गया है क्या? लेकिन उन लोगों का चार्जेस बहुत ज़्यादा है, आई थिंक 2000 है बीएससी नर्सिंग का। लास्ट ईयर मैंने फिल किया था लेकिन एग्ज़ाम देने नहीं गई क्योंकि सिक्किम में सेंटर ही नहीं था। क्योंकि फिर मैं भी चेक करती हूँ, मैं सोच रही थी ना मैं पैरामेडिकल का फॉर्म भरूंगी क्योंकि उसके लिए थोड़ा टाइम बचेगा, नीट के बाद थोड़ा 1 मंथ जितना तो वो टाइम देंगे ही। वो बीएससी नर्सिंग किया था, नर्सिंग छोड़ के आई संगीता। घर में चैन से रहने नहीं देंगे ना, इसलिए पैरामेडिकल उन लोगों को लगेगा तो एटलीस्ट कुछ तो कर रही है ऐसा वाला लगेगा ना, उन लोगों को फील आएगा, तो उसके लिए पैरामेडिकल बीएससी नर्सिंग। वो तो अप्लाई नहीं कर सकता आई थिंक एलेक्स, एम्स में लड़कों के लिए नर्सिंग नहीं है। एलेक्स को ना टेक्नीशियन बनना पड़ेगा शायद। हेलो राशि। राशि तनवार। हाँ एलेक्स, भरूंगी भरूंगी, और तुम भी भरना हाँ। हम दोनों एकदम टीम टीम करके एग्जाम देने जाएंगे। बॉयज के लिए भी है? ओह अच्छा, मुझे लगा कि इसमें नहीं है, नर्सिंग ही। टाइम ड्यूरेशन 20 मिनट कर दिया। हाय शुभम। हाँ मुझे भी ऐसा ही लगा, नर्सिंग में सिर्फ लड़की के लिए है और पैरामेडिकल बाकी जो है वो लड़कों के लिए भी है। दोनों के लिए 2000 है फीस। और आई थिंक हम लोग ना नर्सिंग के साथ साथ पैरामेडिकल भी फील कर सकते हैं। नीट और जेईई। नीट भाई नीट। 1 मिनट हाँ। हाँ हो गया शायद। हेलो आदित्य, यूपीएससी करते हो क्या? अगर डॉक्टर नहीं बनी तो फ्यूचर में करना पड़ेगा शायद। सिविल इंजीनियर ओके। हे तरुण। हाँ फिर हम लोगों के लिए थोड़ा अच्छा है, जो जो फीमेल है उन लोगों के लिए। हाँ है ना एलेक्स? टिल 7th में। अच्छा 7th में तक है लास्ट डेट ऑफ फिलिंग द फॉर्म, या फिर क्या है 7th? Okay. It was something like 277 I guess. 77 ही था शायद। कितने मिनट का स्लॉट रखूं? बोलो कितने मिनट का स्लॉट रखूं? आई थिंक वाईफाई स्टेबल नहीं रहेगा। मैं 12 घंटे सो के उठती हूँ पटेल, मुझसे मांग रहे हो? शांतनु ऐसा तो कोई नहीं है, शेड्यूल नहीं है, मैं पहले भी मेंशन कर चुकी। थर्ड। अच्छा मैं चैट्स ऑफ कर देती हूँ, ओके, वरना तुम लोग नहीं पढ़ पाओगे। गुड लक शुभम, गुड लक एलेक्स। वैसे मेरा टीम अभी नहीं पढ़ेगा आई थिंक, खाना खाने आया होगा ना। ओ नहीं, अभी तो ये लौट के ही आया होगा। गुड लक एवरीवन, जो जो भी पढ़ाई कर रहा है। आई थिंक बाइनरी पढ़ाई कर रहा है, शुभम पढ़ेगा मेरे साथ, और कौन पढ़ेगा? बाकी तो कोई नहीं पढ़ेंगे। हाँ हाँ, सर्वजीता शायद पढ़ी। ओके आदित्य। मैं भारत में रहती हूँ, मुझे क्या पता यूके का टेम्परेचर क्या होगा। पटेल क्या टिप्स, अभी तो क्या ही पढ़ोगे, है ना। अभी कुछ नहीं हो सकता। अगर तुम पढ़ सको तो मेरे साथ पढ़ सकते हो, ताकि तुम्हें एक अच्छा एनवायरनमेंट मिलेगा। यहाँ पर सारे पढ़ने वाले बच्चे होते हैं। अगर तुम ये भी नहीं कर सकते हो तो क्या ही कर सकते। छलांग लगाएंगे ना छत से। एक साल हॉस्पिटलाइज हो जाएंगे, सारे पैसे घर वालों का उड़ा देंगे, फिर वापस आ जाएंगे, फिर से स्टार्ट करेंगे। बच गए तो। मैं क्या करूं? मुझे लग रहा है ना कि नेटवर्क जब स्टेबल नहीं रहेगा तो मुझे नहीं पता चलेगा, लेकिन मैं चैट्स ऑन भी नहीं कर सकते। शुभम, इफ यू आर देयर एंड लिसनिंग, अगर ना मेरा नेट लैग गया तो मुझे पता नहीं चलेगा ना, इसलिए तुम कमेंट कर देना कि मैं नोटिफिकेशन में जाके ना देख लूंगी, फिर वापस से रिफ्रेश कर लेंगे नेट। मैं टाइमर स्टार्ट कर रही हूँ। ओके, स्लॉट 1 स्टार्टेड। Meiosis I involves two sequential cycles of nuclear and cell division called Meiosis I and Meiosis II, but only a single cycle of DNA replication. Meiosis I is initiated after the parental chromosomes have replicated to produce identical sister chromatids as at the S phase. Meiosis involves two sequential cycles of nuclear and cell division called Meiosis I and Meiosis II, but only a single cycle of DNA replication. Two sequential cycle of nuclear and sequential cycle of nuclear under telophase 1, prophase 1, metaphase। आज ना मैं कुछ नहीं कर सकती, इस नेटवर्क का मैं कुछ नहीं कर सकती। प्लीज तुम लोग भी पढ़ो कि वो ये बस नेटवर्क हम लोगों को डिस्ट्रैक्ट करेगा, बाकी कुछ नहीं। प्लीज पढ़ो। Meiosis I, prophase I, metaphase I, anaphase I and telophase I, prophase I, metaphase I and telophase I. Meiosis involves two sequential cycle of nuclear and cell division called meiosis I and meiosis II, but only a single cycle of DNA replication. Two sequential cycle of cell division, nuclear division, single DNA replication, single cycle of DNA replication. Meiosis I is initiated after the parental chromosome have replicated to produce identical sister chromatid at the S phase. Meiosis I is initiated after the parental chromosome have replicated to produce at the S phase. After the parental chromosome have replicated to produce identical sister chromatid at the S phase. S phase ma S phase parental chromosome undergoes replication to produce identical sister chromatid. S phase parental chromosome undergoes replication to produce identical sister chromatids at S phase. At S phase. Yes, phase at S phase. Okay. At S phase. Meiosis meiosis involves pairing of homologous chromosome, pairing of homologous chromosome and recombination between non-sister chromatid of homologous chromosome. Meiosis involves pairing of homologous chromosome. Meiosis involves pairing of homologous chromosome and recombination between non-sister chromatid of homologous chromosome. Pairing of homologous chromosome and recombination of non-homologous chromosome and recombination between non-sister chromatid of homologous chromosome. 4 haploid cells are formed at the end of the meiosis II. 4 haploid cells are formed at the end of the meiosis II. Meiosis involve pairing of homologous chromosome. Here, pairing of homologous chromosome and recombination between non-sister chromatids. Meiosis involves a pairing of homologous chromosome and recombination between non-sister chromatid of homologous chromosome. 4 haploid chromatids are formed at the end of meiosis II. Meiotic event can be grouped under the following phases: meiosis I, meiosis II, meiosis I, prophase and metaphase. Prophase I, metaphase I, anaphase I, and telophase I. Prophase I. Prophase I of first meiotic division is typically longer and more complex when compared to prophase of mitosis. Typically longer and more complex. Okay. Meiosis I. Meiosis I. Meiosis I. Prophase I. Prophase I. Meiosis I, prophase I. Okay. Prophase of the first meiotic division is typically longer, complex, and typically longer and more complex. Complex and longer in comparison to prophase of metaphase I. In comparison to prophase of metaphase I, complex and longer. In comparison to complex and longer in comparison to prophase of mitosis. In comparison to prophase of mitosis. Complex and longer. Complex and longer in comparison to prophase of mitosis. Prophase I. Prophase of the first meiotic division is the phase of mitosis, subdivided into the following 5 stages. Okay, prophase is divided into 5 subphases. So, subdivided into the following 5 phases based on chromosomal behavior: लेप्टोटिन, zygotin, पैकेटिन, डिप्लोटिन and diakinesis. ए शट अप, ये कितना ज़्यादा शोर कर रहा है ना. Subdivided into following 5 phases. 5 phases. PHAGES. Phases. 5 phases. PHAGES. ना, ये तो phases हो गए. Phases. PHASES होता है. PHASES. Phases. Okay, 5 phases. Leptotene. TENE. Leptotene. Then we have zygotin. Ladies हुई है zygotin. Please say packaging. PACHY. Packaging. TEN. Packaging. Leptotin, zygotin, packaging, and finally, little please dance. Diplotene. Diplotene and diakinesis. Diakinesis. D-I-K. D-I-E. Diakinesis. N-E-S-S. Diakinesis. Okay. Leptotin, zygotin, packaging. Leptotin, zygotin, packaging, diplotene, and diakinesis. Leptotin, zygotin, packaging, and diplotene, diakinesis. complex and longer in comparison to prophase of mitosis. Prophase of the first mitotic division is typically longer and more complex when compared to prophase of mitosis. It has been further subdivided into the following 5 phases based on the chromosomal behavior. 5 phases based on its chromosomal behavior. Chromosomal chromosomal behavior. Based on its chromosomal behavior. Okay. Leptotin, zygotin, packaging, diplotene, and diakinesis. Leptotin, zygotin, packaging, diplotene, and diakinesis. Okay. During leptotin stage, the chromosome become gradually visible under the light microscope. During leptotin stage, chromosome becomes gradually visible under light microscope. Leptotin. Chromosome gradually chromosome gradually visible GRADUAL gradually visible under light under light microscope. The compaction of chromosome continues throughout leptotin. Okay, compaction of chromosome continues throughout leptotin. Okay, compaction of continues throughout leptotin. Of chromosome is throughout the leptotin. Chromosome is throughout the leptotin. The compaction of chromosome continues throughout leptotin. This is followed by second stage of prophase I. Okay, ये prophase I है. Prophase I तो है. Leptotin, zygotin, packaging, diplotene, and diakinesis. Chromosome gradually visible under light microscope. Compaction of chromosome continues throughout leptotin. Then zygotin, synaptonemal complex वाला ये. During this stage of chromosome starts pairing together, and this process of association is called synapses. Chromosome starts pairing. CHROMOS. Chromosomes starts pairing. START starts pairing. इसको क्या बोलते हैं? Synapses बोलते हैं. SYNAP. Synapses. Such paired chromosomes are called homologous chromosome. Okay, during this stage, chromosomes starts pairing together. This process of association is called synapses. Such paired chromosomes are called homologous chromosome. Paired chromosomes, chromosomes. Such paired chromosomes are called homologous chromosome. Homologous chromosomes, chromosomes. Homologous chromosomes. Electron micrographs of this stage indicate that chromosome synapses is accompanied by the formation of complex structure called synaptonemal complex. Electron micrograph, GRAPH. Electron micrograph, synaptonemal complex. Complex formed due to the pairing. Complex formed by a pair of synapsed homologous chromosomes is called. Okay. Electron micrographs of this stage indicate that chromosome synapses is accompanied by formation of complex structure called synaptonemal complex. The complex formed by a pair of synapsed homologous chromosome is called bivalent or tetrad. The complex formed by a pair of synapsed homologous chromosome. The complex formed by a pair of synapsed homologous is called a bivalent or tetrad. अच्छा हाँ चटा. A complex formed by a pair of synapsed homologous. Bivalent, valent or tetrad formation, TETRAD, tetrad formation. 1, 2, 3, 4. Four tetrad, Tetrad, tetrad. Tetrad. 2 chromatid, bivalent, okay, bivalent 2 chromatid, 2 CHRO chromatid, 2 chromatid has 4 tetrad, Tetrad. Two chromatid कति रहन्छ नि दुईटा two chromatid कति रहन्छ दुईटा obviously tetrad 1 CHRO chromatid equals to 2 bond, 2 chromatid 4 bond, 4 tetrad, okay. Bivalent or tetrad formation. During this stage, chromosomes start pairing together, and this process of association is called synapses. This process of association is called synapses. Synaptonymal complex. Synaptonymal complex. During this stage, chromosomes start pairing together, and this process of association is called synapses. During this stage, chromosomes start pairing together, and this process of association is called synapses. Such paired chromosomes are called homologous chromosomes. Electron micrographs of this stage indicate that chromosome synapses is accompanied by the formation of complex structure called synaptonymal complex. The complex formed by a pair of synapsed homologous chromosome is called a bivalent or tetrad. However, formation of bivalent or tetrad. The complex formed by a pair of synapsed homologous. The complex formed by a pair of synapsed homologous chromosome is called bivalent or tetrad formation by pairing of homologous chromosome. Bivalent or tetrad formation by pairing of homologous chromosome. Electron micrographs of this stage indicate that chromosome synapses is accompanied by the formation of complex structure. Bivalent or tetrad formation by pairing of homologous chromosome. Graphs of this stage indicate that chromosome synapses is accompanied by the formation of complex structure called synaptonymal complex. Synaptonymal complex. The complex formed by pair of synapsed homologous chromosome is called bivalent or tetrad formation. However, these are more clearly visible at the next stage. The first two stages of Prophase I are relatively short-lived compared to the next stage, that is packaging. Shut up. CHYTEN, packaging. The first two stages of Prophase I are relatively short-lived. The first two stages of Prophase I are relatively short-lived. compared to next stage, that is Packaging. Packaging. Relatively short-lived. The first two stages of Prophase I relatively short-lived compared to the next stage. Two are relatively short-lived. Okay. There's a more clearly bivalent or tetrad. However, there's a more chromosome is more clearly visible. Chromosome. मैं क्या करूँ वाईफाई का आज नहीं अब ना ये खुद ही चलने देते हैं ओके ये मैं ना रिफ्रेश नहीं के बाद खुद ही अगर इसको आना होता है ना तो आ जाएगा मतलब कि जब टाइम आएगा तब ओके आई थिंक जो बचा है ना वो एक शुगर में दूसरा वाला नहीं पता During this stage, okay, the first two stages of Prophase I are relatively short-lived compared to the next stage, that is Packaging. Chromosome dissolve divalent tetrad. Divalent or tetrad more clearly visible. Divalent tetrad more clearly visible. At next stage, however, these are more clearly visible at the next stage. The complex formed by a pair of synapse homologous chromosome is called bivalent or a tetrad. However, these are more clearly visible at the next stage. The first two stages of Prophase I are relatively short-lived compared to next stage, that is Packaging. Packaging during this short-lived, okay. Longer. Goes to phases. First two phases. Prophase of the first meiotic division is typically longer. Prophase of the first meiotic division is typically longer, okay, and more complex when compared to prophase of mitosis. During the stage, the four chromatids of each bivalent chromosome becomes distinct and clearly appears as tetrad. Four chromatid. 2 bivalent equals to 4 chromatid. 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 Bivalent equals to tetrad. Bivalent equals to tetrad. 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 Packaging. During this stage, the four chromatid of each bivalent chromosome becomes distinct and clearly appears as tetrad. The first two stages of Prophase I are relatively short-lived compared to the next stage, that is Packaging. During this stage, the four chromatid of each bivalent chromosome becomes distinct and clearly appears as tetrad. Four chromatids of each bivalent. Of each bivalent, each bivalent. Four chromatids of each bivalent chromosome becomes distinct. Four chromatid of each bivalent becomes distinct. Four chromatid of each bivalent. During this stage, the four chromatid of each bivalent becomes distinct. During this stage, the four chromatid of each bivalent chromosome becomes four chromatid of each bivalent becomes distinct. During this stage, okay, packaging ma distinct and clearly appears as tetrad. Each bivalent becomes distinct and clearly appears as appears as tetrad, and clearly appears as tetrad. Okay. This stage is characterized by the appearance of recombinant nodule, recombination nodules. So, recombination nodules. Recombination nodules. During this stage, the four chromatid of each bivalent chromosome becomes distinct and clearly appears as tetrad. This stage is characterized by the appearance of recombination nodules. The site at which crossing over occurs between non-sister chromatids of the homologous chromosome. Recombinant nodule. CROSSING, crossing over between non-sister chromatids. Crossing over, recombination nodule, crossing over between non-sister chromatids of homologous chromosome. Homologous chromosomes. During this stage, four chromatid of each. The first two stages of Prophase I are relatively short-lived. The complex formed by a pair of synapsed homologous chromosome is called bivalent or tetrad. However, these are more clearly visible at the next stage. The first two stages of Prophase I are relatively short-lived compared to the next stage, that is packaging. During this stage, the four chromatid of each bivalent chromosome becomes distinct and clearly appears as tetrads. This stage is characterized by the appearance of recombination nodules. Recombination nodules, the site at which crossing over occurs between non-sister chromatids of the homologous chromosome. Crossing over is the exchange of genetic material between two homologous chromosomes. Crossing over. Crossing over, exchange of genetic material, exchange of genetic, genetic material, exchange of genetic material, crossing over exchange of genetic material between two homologous chromosomes. Crossing over, exchange of genetic material, exchange of genetic material, exchange of genetic material between two homologous chromosomes. This stage is characterized by appearance of recombination nodule, the site at which the crossing over occurs between non-sister chromatid of the homologous chromosome. Crossing over is the exchange of genetic material between two homologous chromosomes. Between two homologous chromosomes. Crossing over is also an enzyme-mediated process, and the enzyme involved is called recombinase. Crossing over is also an enzyme-mediated process. Enzyme-mediated process. Enzyme involved is called recombinase. Enzyme mediated process. Recombinase. Okay. Crossing over leads to recombination of genetic material. On the two chromosome. recombination of genetic material. Recombination of genetic material. Recombination of genetic material. Crossing over leads to recombination of genetic material on the two chromosomes. Crossing over leads to recombination of genetic material on the two chromosomes. Recombination of genetic material on the two homologous chromosomes is completed by the end of, recombination between homologous chromosome is completed by the end of Paquetin. By end of Paquetin. Leaving the chromosome linked at the site of crossing over. Chromosome linked at the site of crossing over. CHRO. Chromosome linked at the site of chromosome linked at the site of crossing over. Crossing over. At the site of crossing over. Excuse me. Paquetin. Chromosome divalent tetrad, more clearly visible, longer comparison to first two phase. Paquetin. Chromosome divalent tetrad, divalent and tetrad. Full cycle and cell division. Full cycle and cell division. This is the fourth time race. कांड बिलीव एक चैप्टर को 4 बार रिवाइज करने के बाद भी मैं भूल जाती हूँ. हैया Paquetin. Chromosome divalent tetrad, more clearly visible, longer comparison to first two phase. 4 chromatid of each bivalent becomes distinct and clearly appears as tetrad. 4 chromatid of each bivalent becomes distinct and clearly appears as tetrad. Crossing over between non-sister chromatids. Crossing over between non-sister chromatids. Crossing over between non-sister chromatid of homologous chromosome. Recombination nodule and crossing over. It is the exchange of genetic material between two homologous chromosomes. Exchange of genetic material between two homologous chromosomes. Enzyme mediated process. Enzyme is recombinase. Okay. Exchange of genetic material between two homologous chromosomes. Enzyme mediated process. Enzyme recombinase. Recombination of genetic material on the two chromosome. Recombination of homologous chromosome completed by end of the Paquetin. Chromosome linked at the site of crossing over. Crossing over. अस्तगफिरुल्लाह. This is recombinant nodule. Recombination, crossing over. Okay. Recombinase enzyme segments are homologous chromosome but non-sister chromatid. Recombination of HC completed at the end of the Paquetin. You more longer winter. Lipids र zygotin भन्दाखेरि लामो. That is Paquetin. Diplotene. Okay. At the site of crossing over, recombination of homologous chromosome completed. होमोलोगस क्रोमोसोम को रिकम्बिनेसन कंप्लीट हुन्छ, chiasmata formation हुन्छ. The beginning of diplotene is recognized by the dissolution of synaptonymal complex. Okay, diplotene linked at the site of crossing. Fourth one is diplotene. Diplotene, T-E-N-E, diplotene. Diplotene मा के हुन्छ? Dissolution, D-I-S-S-O-L-U-T-N, dissolution of synaptonymal complex. Synaptonymal complex. Dissolution of synaptonymal complex. Diplotene is recognized by the dissolution of synaptonymal complex. Dissolution of synaptonymal complex, diplotene. And the tendency of the recombined homologous chromosome of the bivalent to separate from each other except at the site of chromosomes. Crossover reason. X-shaped structure called chiasmata. In oocyte of some vertebrates, diplotene can last for months or years. The tendency of recombined homologous chromosome. Tendency of recombined homologous chromosome of the bivalent to separate from each other, except at the site of crossover. Recombined homologous chromosome, bivalent. Recombined homologous chromosome. Recombined homologous chromosome. Recombined homologous chromosome at the site of recombined homologous chromosome. मच्छर के बच्चे. Recombined homologous chromosome. Recombined homologous chromosome at the site of recombination. Bivalent. Recombined homologous chromosome of 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 the bivalent to separate from each other. Okay. Chromosome of the bivalent separate. P-E-R-A-T, separate from each other. Homologous chromosome of bivalent separate each other except at the site of crossover. Except of crossover. C-R-O-S-S, cross. Except at the site of crossover. Except at the site of crossover. The tendency of the recombined homologous chromosome of the bivalent to separate from each other. I think so. Crossovers except at the site of crossover. This except structure chiasmata. C-H-I, chiasmata. egg-shaped structure. Egg-shaped structure in oocyte of vertebrates. In oocytes of some vertebrates, diplotene can last for months or years. In oocyte, oocytes, oocytes. In oocyte of some vertebrates. Vertebrates. Vertebrates are over years. In oocyte of some vertebrates, diplotene can last for months or years. मच्छर कितना ज़्यादा काट रहा है. The beginning of diplotene is recognized by the dissolution of synaptonymal complex and the tendency of the recombined homologous chromosome of the bivalent to separate from each other except at the site of crossover. The beginning of diplotene is recognized by the dissolution of synaptonymal complex. Separation of homologous chromosome except at the site of crossover, that is recombination. Egg-shaped structure called chiasmata is seen in this stage or in this phase, that is diplotene. In oocyte of some vertebrates, diplotene can last for months to years. The egg-shaped structure are called chiasmata in oocyte of some vertebrates. Oocytes of some vertebrates. The diplotene can last for months or years. Years. The final stage of meiotic prophase I is diakinesis. This is marked by terminalization of chiasmata. Diakinesis. Last phase. Diakinesis. Final stage of meiotic prophase I. Diakinesis. Final stage of meiotic prophase I. 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 Diakinesis. Final stage of meiotic prophase I. This is marked by the terminalization of chiasmata. Terminalization of chiasmata. 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 Terminalization of chiasmata, okay. The beginning of diplotene is recognized by the dissolution of synaptonymal complex and the tendency of the recombined homologous chromosome of the bivalent to separate from each other except at the site of crossover. The egg-shaped structure are called chiasmata. In oocyte of some vertebrates, diplotene can last for months or years. The final stage of meiotic prophase I. The final stage of meiotic prophase I is diakinesis. It is marked by the terminalization of chiasmata. During this phase, fully condensed and the meiotic spindle is assembled. Chromosomes are fully condensed. Terminalization of chiasmata. Chromosomes are fully condensed. Chromosomes are fully condensed. Chromosomes are fully condensed, and the meiotic spindle is assembled to prepare the homologous chromosomes for separation. Meiotic spindle is assembled. Meiotic spindle. Meiotic spindle. Spindle is assembled. Meiotic spindle is assembled to prepare the homologous chromosome for separation. Meiotic spindle is assembled to prepare during this phase the chromosomes are fully condensed and the meiotic spindle is assembled. The meiotic spindle is assembled to prepare the homologous chromosome. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosome for separation. During this phase, the chromosomes are fully condensed. Meiotic spindle is assembled to prepare the homologous chromosome for separation. Meiotic spindle is assembled to prepare the homologous chromosomes for separation. Homologous chromosome for separation. Okay. Homologous chromosome को separation हुन्छ अलग अलग मान्छ नि त. To prepare the homologous chromosome for separation. By the end of diakinesis, the nucleolus disappears and the nuclear envelope also breaks down. Diakinesis represents transition to metaphase. It represents transition to metaphase. Diakinesis represents transition to metaphase. Okay. To prepare homologous chromosome for separation. End of diakinesis. End of diakinesis. By the end of diakinesis. End of diakinesis. Disappearance of nucleolus, right? Disappearance of nucleolus. End of diakinesis. Disappearance of nucleolus. Nuclear envelope of nucleus. Nuclear envelope. Disappears. DASC. PPEARANC. Disappearance nucleolus. Disappearance of by the end of diakinesis, the nucleolus disappears and the nuclear envelope also breaks down. Diakinesis represents Initiation of metaphase. Transition to metaphase represent transition to metaphase. मैं पानी लेके आती हूँ, ओके? मुझे थोड़ा प्यास लगा है, ऐसा मुझे लग रहा है. Diplotene. Okay. Meiosis I. Meiosis. Key feature of meiosis: two sequential cycle of nuclear and cell division. Meiosis I and Meiosis II. Two sequential cycle of nuclear and cell division. Two sequential cycle of nuclear and cell division. Meiosis I, Meiosis II. One cycle of DNA replication. दुईपल्ट के हुन्छ सेल डिभिजन, दुईपल्ट हुन्छ न्यूक्लियर डिभिजन पनि दुईपल्ट हुन्छ. DNA replication एकपल्ट हुन्छ. S phase मा replicate गर्छ. Meiosis I starts at S phase replication. Meiosis I starts at S phase replication. Replication of parent chromosome S phase मा हुन्छ. Identical sister chromatid. Replication of parent chromosome S phase identical sister chromatid. Replication of parent chromosome S phase identical sister chromatids. Pairing of homologous chromosome. Pairing of homologous chromosomes. Recombination between non-sister chromatid of homologous chromosome. 4 haploid cells at the end of meiosis II. Key feature of meiosis: दुईपल्ट nuclear division, दुईपल्ट cell division, that is two sequential cycle of nuclear division and cell division occurs, but only one third one is DNA replication, single cycle of DNA replication. Meiosis I starts at the Meiosis I starts when parent chromosome replicates. Parent chromosome replicates into identical sister chromatids. S phase one is replication of parent chromosome. S phase identical sister chromatids. Identical sister chromatids. Okay. Then fourth point. ना ये तो दो ही हुआ तो भी. Pairing of homologous chromosome. Non-sister chromatids. Okay. Pairing of homologous chromosome. Recombination between non-sister chromatid of homologous chromosome. Excuse me. 4 haploid cells are produced. 4 haploid cells are produced at the end of meiosis II. ये लोगों को भी एग्जाम है ना, मेरी रूममेट्स का मेरी पहले वाली रूममेट्स का. वो लोग फिर भी व्हाट्सएप में पता नहीं क्या क्या कर रहे हैं. नोटिफिकेशन का टुंग टुंग आ रहा है. व्हाट्सएप ग्रुप पे कुछ तो कर रहे हैं वो लोग. 4 haploid cell at the end of meiosis II. Chromatid chromosomes, 2 chromatids, chromosome or data chromatid से छोड़ दे. Okay, homologous chromosome, non-sister chromatid of homologous chromosome, recombination between non-sister chromatid. Okay, meiosis I, prophase I. Prophase I, complex and longer in comparison to prophase of mitotic division, mitosis बंदम भयो. Complex and longer in comparison to prophase of mitosis. Prophase I, complex and longer in comparison to prophase of mitosis. 5 phases based on the chromosomal behavior. Leptotin, zygotin. Okay, complex. Complex and longer in comparison to prophase of mitosis. Complex and longer in comparison to prophase of mitosis. 5 phases based on its chromosomal behavior. 5 phases based on its chromosomal behavior. 5 phases based on its chromosomal behavior. Leptotin, zygotin, pakitin, diakinesis, and diplotene. Leptotin leptotin के हुन्छ त condensation of chromosome gradually visible under light microscope. Chromosomes gradually visible under the microscope. Compaction, okay, condensation नहीं बोलते से compaction of chromosome continues throughout the leptotin. पता नहीं शायद. मेरा नेटवर्क तो सही है ना, क्योंकि मैं चेक भी कर रही हूँ ना, वाईफाई से कनेक्टेड है. मैं ज़ूम करके चल रही हूँ कि सही है. Prophase I, complex or longer range की leptotin. Chromosomes gradually visible under light microscope. Chromosome gradually visible under light microscope. Compaction of chromosomes continues throughout the leptotin. Chromosomes gradually visible under light microscope. Compaction of chromosomes continues throughout the leptotin. Compaction of chromosome continues throughout leptotin. Then we have zygotin. Chromosomes start spearing up in zygotin. Chromosome starts pairing, यसलाई के भन्छ synapses भन्छ, यसलाई के भन्छ synapses. Such pair chromosome of homologous chromosome. Electron micrograph मा synaptonymal complex देखिन्छ. Synaptonymal complex homologous chromosomes. Crossover भन्दैन, यसलाई के भन्छ भन त? Pairing भन्छौँ. Pairing of chromosome, synapses भन्छ यसलाई. Pairing of chromosome लाई synapses भन्छ. Homologous chromosome को बीचमा pairing हुन्छ. Synaptonymal complex को formation हुन्छ. Bivalent or tetrad formation. Each bivalent has 4 tetrads, 4 chromatids, also called tetrad. Pakitin, leptotin र zygotin भन्दा लामो हुन्छ. Okay, excuse me. इस मच्छर को मैं मार पाऊँ ना तो मैं खुशी हो जाती हूँ, लेकिन मार ही नहीं सकती. Pakitin is longer than leptotin and zygotin. Pakitin longer than it lasts longer than zygotin and leptotin. Okay. Chromosomes divalent tetrad more clearly visible. Pakitin chromosome divalent tetrad more clearly visible. More या फिर longer भन्दा पनि भयो. Longer comparison to first two phases. 4 chromatid of each bivalent becomes distinct and clearly appears as tetrad. 4 chromatid of each bivalent becomes distinct and clearly appears as tetrad. Recombination nodule crossing over between non-sister chromatids. Crossing over between non-sister chromatids of homologous chromosome. Crossing over, exchange of genetic material between two homologous chromosome of non-sister chromatids. Enzyme mediated process. The enzyme required is recombinase. Enzyme mediated process. Enzyme required is recombinase. Recombinase of genetic material recombination completed by end of Packetin. Recombination of homologous chromosome completed by the end of Packetin. Chromosomes link to inside at the site of crossing over. Chromosome link at the site of crossing over. मेरा वाईफाई गया ना फिर से, छोड़ो, लेकिन मैं इसको रिफ्रेश नहीं करती, क्या ही करूँ, 10 बार जाता है. Chromosome linked at the site of crossing over. Diplotene. Dissolution. Diplotene. Dissolution of synaptonymal complex. Dissolution of synaptonymal complex is marked by diplotene. Recombined homologous chromosome of bivalent. Bivalent separate from chromosomes each. Separate from each. Dissolution of diplotene. Dissolution of synaptonymal complex. Recombined homologous chromosome of bivalent separate from each other except at the site of crossover. Except at the site of crossover. Chiasmata. Accept structured. Structure. Accept structure. Accept structure. Diplotene. Dissolution of synaptonymal complex. Dissolution of synaptonymal complex. Recombined homologous chromosome of bivalent separate from each other. Recombined homologous chromosome of bivalent separates from each other. Bivalent separates from each other except at the site of crossover. Dissolution of synaptonymal complex, that is in diplotene, right? Yes, diplotene phase. Dissolution of synaptonymal complex. Recombined homologous chromosome of bivalent separates except at the site of recombination. Except at the site of recombination. Except at the site of recombination vanambayo, crossover vanambayo. Chiasmata. Except structures are formed. That is chiasmata. Okay. In oocyte of some vertebrates, diplotene can last for a few months to many years. Several years. Some words are years. Oocytes of some vertebrates. Diplotene. Oocyte of some vertebrates. Oocyte of some vertebrates. Oocyte of some vertebrates. Dissolution of synaptonymal complex. Recombined homologous chromosome of bivalent separates from each other except at the site of crossover. Chiasmata. X-shaped structure in oocyte of some vertebrates last for months or years. Diakinesis. Final stage of meiosis. Prophase I. Termination of chiasmata. Chromosomes are fully condensed. Meiotic spindle is assembled to prepare homologous chromosome for separation. End of diakinesis. Disappearance of nucleolus and okay. Final stage of meiotic prophase I is diakinesis. Terminalization of chiasmata occur. Chromosomes are fully condensed. Meiotic spindle. Formation of meiotic spindle. Meiotic spindle is assembled to prepare homologous chromosome for separation. To prepare homologous chromosome for separation. Meiotic spindle is assembled. Meiotic spindle is assembled. Meiotic spindle is assembled to prepare homologous chromosome for separation. Homologous chromosome separation को लागि meiotic spindle के गर्नुपर्छ, assemble हुनुपर्छ. Terminalization of chiasmata. Chromosomes are fully condensed. Meiotic spindle is assembled to separate the homologous chromosome. End of diakinesis. End of diakinesis मा दुईटा कुरा हुन्छ कि के हुन्छ भन्दा end of diakinesis मा end of diakinesis. Disappearance of nucleolus and nuclear membrane. Disappearance of nucleolus and nuclear membrane. Disappearance, right? Yes, it is disappearance or disappears. The nucleolus disappears and the nuclear envelope also breaks down. Nuclear envelope breaks down. Okay. Diplotene is the phase that represents the transition to metaphase. Diakinesis. Diakinesis is the phase that represents transition to metaphase. Transition to metaphase. Transition to metaphase को ले बताउँछ diplotene ले. Metaphase I. The bivalent chromosome align on the equatorial plate. The bivalent chromosome Bivalent chromosome align on the equatorial plate. Equatorial plate में bivalent chromosome के हुन्छ arrange हुन्छ. The microtubules from the opposite pole of the spindle attached to the kinetochore of homologous chromosome. Attached to the kinetochore of homologous chromosome. The microtubules from the opposite pole of the spindle attached to the kinetochore of homologous chromosome. Anaphase 1. The homologous chromosome separates while sister chromatids remains associated with their centromere. Alignment. Bivalent chromosome align on the equatorial plate. The microtubules from the opposite poles of the spindle. The microtubules from the opposite pole of the spindle attached to the kinetochore of homologous chromosome. Equatorial plate में bivalent chromosome के गर्छ arrange गर्छ या फिर align गर्छ. Okay. Equatorial bivalent chromosomes align on the equatorial plate. The microtubules from the opposite pole of the spindle attached to the kinetochore of homologous chromosome. The final stage of meiotic phase 1 is diakinesis. This is marked by the terminalization of Chi's matter. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosome for separation. By the end of diakinesis, the nucleolus disappears and the nuclear envelope also breaks down. Diakinesis represents transition to metaphase. By the end of diakinesis, the nucleolus disappears and the nuclear envelope also breaks down. Diakinesis represents transition to metaphase. Metaphase 1. Bivalent chromosome aligns to the equatorial plate. The bivalent chromosomes align to the equatorial plate. The microtubules from the opposite poles of the spindle attached to the kinetochore. Opposite pole of the spindle. The microtubules from the opposite pole of the spindle attached to the kinetochore of homologous chromosome. Anaphase. The homologous chromosome separates. Okay, here. Homologous chromosome, not chromatid. The homologous chromosome separates while sister chromatids remains associated with the centromere. Homologous chromosome separate. Homologous chromosome separate on this chromosome separate on this. Homologous chromosome separates, but sister chromatids are associated at their centromeres. The bivalent chromosomes align to the equatorial plate. The bivalent chromosomes align to the equatorial plate. Anaphase 1. Telophase 1. The nuclear membrane and the nucleolus reappears. The nuclear membrane and the nucleolus reappears. Cytokinesis follows, and this is called as the dyad of cell. Figure 10.3. Telophase. Okay. Nuclear membrane reappears. Nuclear membrane reappears as well as nucleolus reappears. Nuclear membrane and nucleolus reappears. Cytokinesis follows, and this is called as dyad of cell. Cytokinesis follows, and this is called as dyad of cells. Although in many cases the chromosome do undergo some dispersion. Okay, dyad of cell की बनी रहती है dyad of cells. Although in many cases the chromosomes do undergo some dispersion, they do not reach the extremely extended state of the interphase nucleus. They do not reach the extremely extended state of the interphase nucleus. The stage between the two meiotic division is called kinesis and is generally short-lived. There is no replication of DNA during interkinesis. Interkinesis is followed by prophase II a much simpler prophase than prophase one. अच्छा, तो ये तुम्हें करना होगा telophase वाला. Telophase. Nucleolus reappears. Cytokinesis follows. This is called dyad of cell. इसमें क्या इंसान था? Nucleolus and nuclear membrane. Nuclear envelope. Nucleus and nuclear envelopes. Nucleolus. Nucleolus and nuclear envelopes. Envelope reappears. Cytokinesis occur. Cytokinesis occurs. What's the CURS? Occurs. Cytokinesis occurs. Dyad of cell. Nucleolus and nuclear envelope. Nucleolus and nuclear envelope. The nuclear membrane and nucleolus reappears. Cytokinesis follows. Dyad of cell. Okay. Although in many cases the chromosomes do undergo some dispersion, they do not reach the extremely extended state of the interphase nucleus. Prophase, metaphase. Interkinesis. Yes. The stage between two meiotic division. Stays between two meiotic division is called interkinesis and is generally short-lived. There is no replication of DNA during interkinesis. No replication of DNA during interkinesis. No DNA replication. Astaghfirullah. During this interkinesis. Interkinesis is followed by Prophase II, a much simpler prophase than Prophase I. Interkinesis is followed by Prophase II. Meiosis II, Prophase II, Meiosis II. Nuclear membrane and nucleolus reappears. Cytokinesis follows, and this is called as dyad of cell. Although in many cases the chromosome do undergo some dispersion, they do not reach the extremely extended state of interphase nucleus. They do not reach the extremely extended state of interphase nucleus. Interphase nucleus. okay, ma. It's okay, Ima. G1S and G2. G1S, G2. G1S, G2. Interface. G1S, G2. Interface. They do not reach the extremely extended state of interphase nucleus. The stage between two meiotic division is called interkinesis. Interkinesis, and is generally short-lived. Interkinesis stays between two meiotic division. State between two meiotic division is called interkinesis. Generally interkinesis is short-lived. There is no replication of DNA during interkinesis. Interkinesis is followed by Prophase II, which is much simpler in comparison to Prophase I. बस इतना ही तो है. अभी भी 10 मिनट बचा है और मुझे नींद आ रही है. The stage between meiotic division is called interkinesis and is generally short-lived. Interkinesis is generally short-lived. सोना तो नहीं चाहिए मुझे. Interkinesis is generally short-lived. There is no replication of DNA during interkinesis. It is followed by Prophase II. Prophase II. Meiosis II is initiated immediately after cytokinesis, usually before the chromosome has fully elongated. Usually before the chromosomes. Prophase I, metaphase I, anaphase I. This is telophase I. Okay, stages of meiosis. क्यों नींद आ रही है? मुझे क्या पता क्यों नींद आ रही है. telophase I. Prophase II. Meiosis II is initiated immediately after cytokinesis, usually before the chromosome have fully elongated. In contrast to Meiosis I, Meiosis II resembles a normal mitosis. The nuclear membrane disappears by the end of Prophase II. 10.4. The chromosomes again become compact. At this stage, the chromosome aligns at the equator, and the microtubules from the opposite pole of the spindle gets attached to the kinetochore of sister chromatids. Okay, homologous chromosome bandite sister chromatids bandis. Okay, Meiosis II and the sister chromatids. Anaphase II. It begins with the simultaneous splitting of the centromere. It begins with the simultaneous splitting of the centromere. Excuse It begins with the simultaneous splitting of the centromere of each chromosome, which was holding the sister chromatid together, allowing them to move towards opposite poles of the cell, shortening of microtubules attached to kinetic pole. Telophase II. Meiosis ends with the telophase II, in which the two group of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of cell, that is, four haploid daughter cells. Significance of meiosis. Meiosis is the mechanism by which conservation of specific chromosome number of each species is achieved. Conservation of chromosome number. Conservation of chromosome number of each species, okay, achieved across generation is in sexually reproducing organism. Even though the process per se paradoxically results in the reduction of chromosome number by half, it also increases the genetic variability. Okay, increases genetic variability. Conserve chromosome number. Conserve chromosome number increases the genetic variability in the population of organism from one generation to the next. Variation is very important for the process of evolution. Variation is very important for the process of evolution. Genetic increases the genetic variability in the population of organisms from one generation to the next. Conservation of chromosome number. Increase the genetic variability. ऐसा ना हो, मैं नहीं चाहती ऐसा हो. Stages of meiosis II. Prophase, metaphase, anaphase, telophase हो गया. अब रहा हम लोग का EM फेज. पहले पढ़ने से पहले ये पढ़ो. Cell division is very important process in all living organisms. During the cell division of a cell, DNA replication and cell growth also takes place. During the division of cell, cell division को लागि के हुँदाखेरि के डिवाइड हुन्छ चाहिँ DNA replicate हुन्छ र cell को growth पनि हुन्छ. Cell division is very important process in all living organisms. During the division of cell, DNA replication and cell growth also takes place. All these processes, that is cell division, DNA replication and cell growth, hence have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. The sequence of event by which a cell duplicates its genome, synthesize the other constituents of the cell, and eventually divide into two daughter cells is termed as cell cycle. Wait, the sequence of event by which a cell duplicates its genome, synthesize the other constituent of the cell, and eventually divides into two daughter cells is termed as cell cycle. Although cell growth in terms of cytoplasmic increase is a continuous process. Cell growth in terms of cytoplasmic increase is a continuous process. DNA synthesis occurs only during one specific stage. DNA synthesis occurs only during one specific stage in the cell cycle. The replicated chromosomes DNA are then distributed. The replicated chromosome DNA. छोड़ो ना. Chaoima, please concentrate. The replicated chromosomes DNA are then distributed to daughter nuclei. The replicated chromosomes are distributed to daughter nuclei by a complex series of events during the division. These events are themselves under genetic control. Phases of cell cycle. A typical eukaryotic cell cycle is illustrated by human cell in culture. Typical eukaryotic cell cycle. Okay. These cells divides once in approximately every 24 hours. Okay, human cells divide once in approximately 24 hours. 24 hours, one cell division in humans. However, the duration of cell cycle can vary from organism to organism and also from cell type to cell type. Yeast, for example, can progress through the cell cycle in only about 90 minutes. Yeast 90 minutes, human 24 hour, and then E. coli 20 minute, 90-minute yeast. The cell cycle is divided into two basic phases: interphase, M phase, mitosis phase. Interphase and mitosis phase, M phase, mitosis phase. G1S, G2. G1S, G2. The diagrammatic view of cell cycle indicating formation of two cell from one cell. Formation of two cell from one cell. Okay. G0 pani interface. G0, G1S and G2. G0, G1S, G2. S, G2, GS2. G1S, G2. G1S, G2. GSG ऐसे करके याद करना पड़ेगा इसको. मैं हमेशा भूल जाती हूँ इसकी ऑर्डर. मुझे G2 पहले आएगा कि S पहले आएगा पता ही नहीं चलता. G1S पहले आएगा. ओके, S. कैसे याद कर सकते हैं इसको हम? G2 को ऐसे कर याद कर सकते हैं हम. एक गीतू नाम की लड़की है, वो पोकेमॉन की, वो ऐश है ना, एस से बाद में आएगी क्योंकि एस के पास पोकेमॉन है तो वो ताकतवर है. ऐसा करके याद कर सकते हैं ना. G1, then S, then G2, then M phase. Prophase, metaphase, anaphase, telophase, and finally cytokinesis. Prophase, metaphase, anaphase, telophase, and cytokinesis. Diagrammatic view of cell cycle indicating formation of two cells from one cell. Okay. Interphase and M phase. Mitosis phase. The M phase represents the phase when the actual cell division or mitosis occurs, and the interphase represents the phase between two successive M phase. Interphase represents the phase between two successive mitosis. M phase है ना. आगे कहते हैं interkinesis between two meiosis. Interkinesis. Okay. It is significant to note that in the 24-hour average duration of cell cycle of a human cell, cell division proper lasts for only about an hour. The interphase lasts more than 95% of the duration of the cell cycle. Interphase lasts more than 95% of the duration of cell cycle. The M phase starts with the nuclear division. M phase starts with nuclear division. M phase starts with nuclear division, corresponding to the separation of daughter chromosomes. Separation of daughter chromosome. Karyokinesis. Karyokinesis, separation of daughter chromosome. Separation of daughter chromosome, karyokinesis, usually ends with the division of cytoplasm. Cytokinesis. Okay. Karyokinesis, separation of daughter. Karyokinesis. Karyokinesis. Separation of daughter chromosome. Separation of daughter chromosome, karyokinesis, and usually ends with the division of cytoplasm. Cytokinesis. The interphase, though called resting phase, interphase is also called a resting phase. Okay, resting phase, interphase, 95%. Okay, is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication. Interphase में DNA replication? हाँ, तो S phase में होगा ना, interphase में ही तो आएगा. In the time during which the cell preparing for interphase is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication. By undergoing both cell growth and DNA replication. एक बात बोलूं, हम लोगों को ना ब्रेक का टाइम चलने वाला था ना ये, मतलब कि हम लोगों ने फर्स्ट लर्ड कंप्लीट किया है और एक्स्ट्रा 11 मिनट समथिंग कितना भी पढ़ लिया है, क्योंकि अब ना ये ना ऊपर की तरफ इंक्रीज हो रहा है टाइमर. देखो तो पता नहीं टाइमर को क्या हुआ है. कैन यू सी? सेकेंड वाला काटा ये ना इंक्रीज हो रहा है. मैं इसको पॉज कर देती हूँ. The M phase starts with the nuclear division corresponding to the separation of daughter chromosome, karyokinesis, and usually ends with the division of cytoplasm, cytokinesis. The interphase, though called the resting phase, is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. The interphase, though called resting phase, is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in an orderly manner. The interphase is divided into three further phases: G1, S, and G2. G1, S, and G2. Gap 1, synthesis, and gap 2 phase. Gap 1 phase, synthesis phase, and gap 2 phase. Okay, gap 1, gap 2. Gap 2 से पहले synthesis phase. G1 phase corresponds to the interval between mitosis and the initiation of DNA replication. Between mitosis and initiation of DNA replication, during G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. metabolically active and continuously grow but do not replicate its DNA. S or synthesis phase marks the period during which DNA synthesis or replication takes place. Okay, during this time the amount of DNA per cell doubles. Okay. If the initial amount of DNA is denoted as 2C, then it increases to 4C. However, there is no increase in the chromosome number. Chromosome number ma ke increase undena. If the cell had there is no increase in chromosome number. Chromosome number increase कर देना सिर्फ DNA amount of DNA मते increase करता. Okay. If the cell had diploid or 2n number of chromosome at G1, even after S phase, the number of chromosome remains the same. In animal, during S phase, DNA replication begins in the nucleus. The centriole duplicates in cytoplasm. In animal, during S phase, DNA replication begins in nucleus. DNA replication begins in nucleus. Centriole duplicates in cytoplasm. During G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. Some cells in the adult animals do not appear to exhibit division. Example, heart cell do not appear to exhibit the division. Heart cells divide do not appear to exhibit division. Some cells in the adult animals do not appear to exhibit division. Example, heart cell, and many other cell divides only occasionally as needed to replace cells that have been lost because of injury or cell death. These cells that do not divide further exit. These cells that do not divide further exit Z1 phase to enter an inactive stage called quiescent stage G0. Inactive stage called quiescent stage at G0 of the cell cycle. Cell in this stage remains metabolically active but no longer proliferate unless called on to do so, depending on the requirement of the organism. Metabolically active, no longer division. Cell division होंगे ना, proliferate unless called on to do so. G0, quiescent stage, inactive stage. In animals, mitotic cell division is only seen in the diploid somatic cell. In animals, mitotic cell division is only seen in diploid somatic cell. However, there are few exceptions of this where haploid cell divide by mitosis. For example, male honeybees. Oh, अच्छा ये दिया है तो यहाँ पर मैंने नहीं पढ़ा था. I think ये old book पे नहीं दिया है. लेकिन क्लास 12 में जाके ये पढ़ना ही तो पड़ेगा. इससे अच्छा भी देख लेते हैं. Against the plant can show mitotic division in both haploid cell and diploid. हाँ यहाँ पर नहीं दिया है, ओल्ड बुक पे नहीं दिया है. देखो ये पैराग्राफ में, न्यू बुक में दिया है इसमें. In animals, mitotic cell division is only seen in diploid somatic cell. However, there are few exceptions where haploid cells divide by mitosis. Haploid cell divides by mitosis. For example, male honeybees मतलब से haploid cell divides. अगर ये आता भी मैं कर लेती क्योंकि मुझे पता था. ये उसमें भी है ना, वो वाला चैप्टर कौन सा था, प्रिंसिपल ऑफ इनहेरिटेंस. Haploid cell divide by mitosis, for example, male honeybees. Against this, the plant can show mitotic division in both haploid and diploid cell. Plants can show both haploid and mitotic division in both haploid and diploid cell. For your recollection of example of alteration of generation in plant. Chapter 3. Animal plant species and stages at which mitosis is seen in haploid cell. The diploid cells divide by mitosis. Male honeybee. Against this, plant can show mitotic division in both haploid and diploid cells. Is only seen diploid somatic cell. Plant against this, the plant can show mitotic division in both haploid and diploid cells. From your recollection of example, alteration of generation in plants. Sixteen chromosome in each cell. Can you tell how many chromosomes will the cell have at G1? 16 chromosomes. 16. After M phase also, what will be the DNA content of the cell at G1? To see. 4C. If the content after M phases, 2C. तो 29% ही तो है. If the total amount or number of chromosome is 16 in G1 phase, what will be the number of chromosome in M phase? The number of chromosome in M phase gives the number of chromosome in G1. Okay. The number of chromosome in M phase given the number of chromosome in G1 phase. मतलब इसमें 16 ही होगा. The number of chromosome in M phases. Number of chromosome. G1. 2C, 4C. M phases 2C. If the DNA content of the cell at G1 is 2C, what will be the content of DNA after M phase? Content is 2C in G1 phase, meaning that one set of chromosome, it will remain 2C after M phase. Mitosis, the cell divides and DNA equally into the right end. अच्छा. Gap to 4C, M phase 2C. 2C. DNA content divides. DNA 4C divides equally ब्रेक लेंगे, ओके 10 मिनट का। इसको ना मैं एक बार ये कर देती हूँ, वरना ये फिर से उल्टा दौड़ने लगेगा। अजीब है। टाइम कितना है? 10 मिनट का ब्रेक बचा। स्लॉट 1 completed as load is slot 1 completed. Break of 10 minutes. 10 minutes. ये लो, रिप्लेस कर दिया मैंने। Normal, average. I think that was average. Average. मैं पानी लेके आती हूँ ना, मुझे ज़्यादा नींद लगेगा अगर मैं पानी नहीं लेके आई तो। 10:33, 10:33, 11 भी नहीं हुआ है और मुझे नींद आ रहा है। हाँ, ये वाला हमने थोड़ा ज़्यादा पढ़ा क्योंकि टाइमर बीप नहीं किया ना, हम लोगों ने एक्स्ट्रा पढ़ लिया। आई थिंक 12 मिनट एक्स्ट्रा पढ़ा। तो एक स्लॉट में ही हो गया। हाँ, ये स्लॉट लंबा रखा ना, इसलिए ऐसा लग रहा है तुम्हें। 6 AM तक मैं तो सो जाऊंगी, ऐसा मुझे लग रहा है। वैसे मेरी तबीयत सही नहीं है ना, आज तो फिर भी बेटर फील कर रही हूँ, कल तो मरने वाला जैसा हो गया था। फिर मुझे ना लाइफ ऑन करके इस तरह सोना पड़ेगा, और इस तरफ मैं सोऊंगी नहीं क्योंकि वो कैटरपिलर वापस आएगा। तो देखो मेरा हाथ अभी भी ठीक नहीं हुआ है तब मैं तुम लोगों की वजह से यहाँ पर सो रही थी, लेकिन अब तो मैं नहीं सोऊंगी, मैं मेरे ही बेड पे सोऊंगी। और मेरा हाथ भी ऐसा हो गया गंदा वाला सा। कैन यू सी यहाँ पर कैटरपिलर ने ना अपना हेयर यहाँ पर डाल दिया था, वो मेरे बेड पे आया था ना, मुझे पता ही नहीं चला। फिर मुझे बहुत ज़्यादा डरता है तो मैंने देखा भी नहीं। वो मेरा बचपन से एक फोबिया टाइप है ना, क्योंकि वो जेनेटिक है, मेरे बाबा को भी था। लाइक मेरी अम्मा थोड़ा स्ट्रांग है, उन्होंने मैंने जाके बताया ना मामा मेरे हाथ में ऐसा हो गया था और मेरे पैर में भी ऐसा कुछ आ गया है हेयर टाइप। तो उन्होंने बोला कि कैटरपिलर ही होगा, अच्छे से नहीं देखा क्या तूने, ऐसा करके बोला ना। मैंने बोला कि मैं नहीं देख सकती। फिर अम्मा आई और उन्होंने देखा कि उन्होंने बोला ओ इतना बड़ा है ऐसा करके फिर मैं देखने नहीं आई, मैं चिल्लाई भी क्योंकि मुझे ज़्यादा डर लगता था ना। फिर अम्मा ने इसको मार दिया शायद। फिर दूसरे दिन ना अम्मा बोल रही थी कि एक और उनके रूम में एक कैटरपिलर आया है, और वो तो इतना बड़ा था कि अम्मा को ही डर लगा। हाँ बंद कर दूंगी ऑब्वियसली। हाँ तुमने सही बोला सुदीप, अभी मैं पानी लेने भी जा रही हूँ और मैं बाहर जाऊंगी भी। आ, सुदीप की बात सुन लेते हो, ओके मैं जा रही हूँ, बाय बाय। मुझे क्योंकि ना पानी गर्म करके लेके आना है और खूब सारे पानी पी के ना रात भर नींद को भगाना है। अगर हम लोगों ने पानी नहीं पिया तो हम लोगों को कॉफ़ी पीना पड़ेगा, जो कि मैं नहीं पी सकती अभी तबीयत की वजह से। और पानी पिया तो नींद नहीं आएगी। अभी जा रही हूँ, ओके ज़्यादा टाइम नहीं बचा, 6 मिनट ही बचा है, मैं वापस आ जाऊँगी जल्दी 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 वापस आ जाऊँगी। अरे थैंक यू सुदीप। एग्जाम के लिए मुझे बेस्ट बोल दिया। श्री सोते कब हो? श्री दिन में सोता होगा शायद। स्त्री हो गया, ये गलत बात है। Don't you? नाइन्टी मिनट का स्लॉट ज़्यादा हो जाएगा बाइनरी। मुझे भी तो बैठने के लिए ज़्यादा मुश्किल पड़ेगा ना। रील में सिक्किम से हूँ। Don't you get distracted while conversation? We don't converse it while studying. This is our break time. We usually turn off the comment section or chat section or chats, whatever. We disable it. 100 मिनट ना 90 मिनट ही ज़्यादा हो रहा है। आ गए 100 मिनट। मैं 60 मिनट के लिए वोट करती हूँ, बाइनरी 90 मिनट के लिए वोट करता है और शुभम 100 मिनट के लिए। बाकी कोई? श्री आर यू देयर? तुम बोलो। श्री जो बोलेगा, जो नंबर बोलेगा वही जीतेगा। श्री आर यू देयर? आई डोंट थिंक ही इज देयर। आप में से कोई एक ना, मुझे पता है सारे लोग नहीं पढ़ रहे हैं, लेकिन कोई एक नंबर बता दो, 90, 100 और 60। कौन सा वाला? कितने मिनट से? जल्दी जल्दी जल्दी। स्लॉट 2 स्टार्टेड। SLOT स्लॉट 2 स्टार्ट्स। अच्छा स्टार्ट्स। शुभम ने ही बोल दिया, शुभम ने चेंज किया। थैंक यू शुभम। 60 मिनट। तक पढ़ूंगा बोल रहा था श्री। अब चला गया। अरे नहीं, वो बेचारा से मत बोलो ना। श्री अच्छा है, वो पढ़ता है सच्ची में, लेकिन ना वो यहाँ पर नहीं आ सकता लाइव पे क्योंकि वो वीडियो लेक्चर्स फॉलो करता है। हाँ बाइनरी ने भी 60 बोल दिया। ओके थैंक यू। सब लोग 60 बोलने लगे तो 60 ही करते हैं। ओके तुम वापस से आ गए। मैं इसका भूली ही नहीं हूँ। इसका 310 आया था तो मैं इसको भूल ही नहीं पा रही हूँ। अच्छे से मुझे याद है, लेकिन छोड़ो, मैंने पिन कर दिया है ना। वैसे मैंने 70 मिनट का लगा लिया है तो 70 मिनट ही पढ़ते हो। ओके गुड लक शुभम एंड बाइनरी। श्री को भी गुड लक। तीन 310। मैंने चैट्स डिसेबल कर दिया। ओके नाइस। माइटोटिक फेज। मैं टाइमर ऑन कर दे रही हूँ। ओके, अभी तक मैंने टाइमर ऑन नहीं किया है। 70 मिनट का है स्लॉट 2, हालांकि मैंने 60 मिनट करके लिख दिया है, लेकिन 70 मिनट का है। गुड लक एवरीवन। Preparatory phase, resting phase. Actual cell division not observed comprises 3 stages, subphases G1, S phase and G2, G0 phase stage. G1 और here so cycle में चली वैसे G1 phase gap 1, most of the cell organelles duplicates. RNA and protein synthesis, DNA, most of the protein duplicates. Protein organelles, so most of the organelle duplication. RNA and protein synthesis, DNA precursor, excuse me, DNA precursor, nucleotide, amino acid, sugar synthesized. Longer the phase, slower will be the division. Longer the phase, slower will be the division. As phase, synthetic phase, DNA duplication, nucleus, centriole duplication, cytoplasm, histone protein synthesis. Okay, G1 phase, S phase, G1 phase and S phase. Okay, DNA duplication in S phase, sister chromatid duplication, chromosome 1, chromosome X, 1 sister chromatid. If amount of DNA at G1 is 2C, amount of DNA at S will be 4C. Example, if 8 picogram at G1, amount at S is 16 picogram. DNA double means, okay. If the amount of chromosome at the G1 is X, number of chromosome at S remains X. Total chromatid at the G1 is N, then number of chromatid in S is 2N. G2 phase, gap2 phase, tubulin protein synthesis, special state of organelle duplication, chloroplast, mitochondria and Golgi bodies. Okay, RNA and protein synthesis. Okay, G2 लेके गर्छ tubulin protein synthesize गर्छ. G0 phase referred to as quiescent phase. Phase of metabolism, a phase of metabolism in all the cell without undergoing proliferation. Metabolism observed, cell proliferation. Cell proliferation. सुत्नु म चाहिँ सुतेको छुइनँ, आमा त सुत्नु भयो? हजुर। मेरे घर ना रास्ते में ही है ना, तो उधर जाने वाले लोग बोल रहे हैं, मेरे अम्मा को बुला रहे। मैंने बोला कि अम्मा सो रही है, 11 बज गए हैं। पता नहीं क्यों पूछा, मुझे नहीं पता। Metabolism observed, cell proliferation not observed, the condition where cell can exit the cell wall. Metabolism in the cell without undergoing proliferation. Metabolism observed, cell proliferation not observed, the condition where all cell exit. मुझे नहीं जवाब देना था, तूने दे दिया, अब क्या ही कर सकते हैं. Cell proliferation not observed, the condition where cell can exit the cell wall. Cell cycle, the condition where cell can exit the cell cycle. The condition where cell can exit the cell cycle temporarily in deficiency or requirement. It appears into active cell division. Permanently ceases to divide. Nerve cell, even in hard cardiac cells. Mitotic phase, M phase, comprises of two processes: karyokinesis, nucleus division, and cytokinesis, cell division. Karyokinesis, nuclear division. Cell regulation, cell cycle regulated or maintained by certain signalings. ये वाला पोर्शन छोड़ो, ये नहीं दिया है ना चेक पॉइंट वगैरह, ये तो नहीं आएगा शायद, आ भी सकता है. How would daughter cell at the end of mitosis and cytokinesis compared with their parent cell when it was in G1 of the cell cycle? How would daughter cell at the end of mitosis and cytokinesis compared with the parent cell when it was in G1 of the cell cycle? Daughter cell have the same number of chromosomes, same amount of DNA. Mitosis observed by Starberger in plants, Fleming in animals. Observed in plants and Fleming in animals. Term coined by Fleming. Mitosis, okay, consists of two processes: karyokinesis and cytokinesis. Cytokinesis involves two types of cell division in animal cell: poroformation. Poroformation in plant cell, cell plate formation. Karyokinesis involves subphases. Karyokinesis subphases: Prophase I, metaphase I, anaphase I, and telophase. Not one. Prophase, metaphase, anaphase, and telophase. Prophase, metaphase, anaphase, and telophase. Animal cell, poroformation. Plant cell, cell plate formation. Plant cell, cell plate formation in plant. Animal cell, poroformation. Mitosis, division of one cell into daughter cell. Each daughter cell receives equal number of chromosome, which is equal number of chromosome in plant cell. Mitosis, division of one cell into two daughter cell. Each daughter cell receives equal number of chromosome. One cell into two daughter cell. Each daughter cell receives equal number of chromosome, which is equal number of chromosome in parent cell. Parent cell 4 to 6 chromosome. One cell. Daughter cell, second cell, daughter cell. Both have 4 to 6 chromosome. Equational division, mitosis, generally observed in diploid cell in animals and both haploid and diploid cell in plant. Exception diploid animal को diploid cell माते हुँदैन. Exception के हो honeybee, male honeybee. Karyokinesis, one daughter nuclei equals to two daughter nuclei in the same cell contained within the cell. Karyokinesis, one daughter nuclei equals to two daughter nuclei in the same cell contained within the cell. Prophase, first stage, initial event of prophase, chromatin, chromosome condensation. Chromosome condensation appears as entangled fiber, centrioles that was duplicated in the S phase of interphase. Cytoplasm starts moving towards the pole. Centriole starts moving towards the pole in prophase. First initial event of the prophase, chromatin, chromosome condensation appears as entangled fiber, centriole that was duplicated in the S phase of interphase starts moving towards the pole. Elongated mature centriole at G2. Astral rays formation emergence is observed. In the initial prophase, the chromatin fiber appears like ball of wool. Okay, prophase, mitosis को prophase नाम चला. Chromatin fiber appears like ball of wool. Ball of wool, chromatin fiber, referred to as aspirin stage ना. कौन पता नहीं था. Based on astral formation, two types of mitosis. Without astral formation, anastral in plants. With astral formation, astral, amphiastral. Astral or amphiastral in animals. Centriole present. Centriole छ त्यसै कारण के हुन्छ amphiastral या फिर astral हुन्छ. Plant मा astral formation छैन त्यसै कारण के हुन्छ centriole के astriole. Centriole छैन त्यसै कारण anastral हुन्छ. Astral is composed of centrosome with astral rays. Late event of prophase, chromosome condensation, centriole movement poleward, the astral rays formation radiation, the disappearance of nucleolus, Golgi bodies, and the disintegration of nucleus. Disappearance of nucleolus, endoplasmic reticulum, and Golgi body, along with the disintegration of nuclear membrane. Disintegration of nucleus. Prophase. At the completion of prophase, the chromosome condensation, centriole poleward movement, disappearance of nuclear envelope, disintegration, nucleolus, Golgi body disappears. Nuclear envelope disintegrate. Wonderful. It starts with all the chromosomes scattered. Metaphase. It starts with all the chromosomes scattered or placed, observed throughout the cell. It starts with all the chromosomes scattered or placed, observed throughout the cell. Initial metaphase. The chromosomes shortest and thickest. Best phase to observe morphology or chromosomes structure. नहीं ह्यूमन फिजियो वाला पोर्शन ना अब ज़्यादा वो लग रहा है, लेकिन पता नहीं क्या कर सकते शायद. ऊपर का पढ़ती हूँ ना, मुझे ऐसा लगता है कि नीचे वाला पोर्शन भूल जा रही हूँ. छोड़ो, पढ़ते हैं. With all the chromosomes scattered or placed, observed throughout the cell. Initial metaphase. The chromosome shortest and thickest. Best phase to observe morphology of chromosome structure. Centriole produce spindle fiber which connects to kinetochore. Centriole produce spindle fiber which connects to kinetochore. Later metaphase. Process of arrangement of chromosome. Metaphase. Okay, it starts with all the chromosomes scattered or placed, observed throughout the cell. Initial metaphase. The chromosome shortest, thickest. Best phase to observe morphology of chromosome structure. The chromosome shortest and thickest. Best phase to observe morphology of chromosome structure. Metaphase. Centriole produce spindle fiber which connects to kinetochore. Centriole produce spindle fiber. Okay, best phase to observe morphology of the chromosome. Later metaphase. Process of arrangement of chromosome. Metaphasic plate. Spindle fiber. Equatorial plate arrangement. The process of arrangement of chromosome in the metaphasic plate. Congregation. Spindle equator. Spindle fiber. Spindle equator. Later metaphase. Process of arrangement of chromosome. Metaphase plate. Anaphase. Ana मतलब up stage. Anaphase, up stage. Okay. The separation of sister chromatid towards each pole. The separation of sister chromatid towards each pole. Separation is brought by shortening of spindle fiber connected to kinetochore and elongation of interzonal fiber. Shortening of spindle fiber, elongation of interzonal fiber. The separation of sister chromatid towards each pole. Separation is brought by shortening of spindle fiber but elongation of interzonal fiber. Sister chromatid separate होंगे. So आगे meiosis I में homologous chromosome separate होंगे क्यों? Excuse me. Sister chromatid. Centromere division towards the pole. Chromatid are trailing behind. Centromere division towards the pole. Shortening of spindle. Interzonal fiber. As each chromatid are separated from the chromosome, the chromatid aggregate at each pole becomes part of daughter nuclei. Each chromatid. 46 chromosome. Chromatids in G1, 46 chromatids in S, 92. Human cell 46 G1 में 46 chromosome S में अपनी 46 से number of chromosomes same हुन्छ 46 chromatid 92. G2 में 46 chromatid 92. Prophase में same, metaphase में same, anaphase में 46 chromosome 46 chromosome each. Ninety-two chromatids divide. 92 chromatids divide equally. Anaphase exactly same number of chromatid as G2. Telophase reverse the process. Prophase chromosome. Decondensation. Centriole disappears. Spindle fiber disappearance. Nuclear membrane reappears. Telophase chromosome. Chromatid decondensation. Reverse of prophase. Okay. Chromosome decondensation. Centriole disappears. Centriole disappears. Reappear. Centriole disappear, spindle fiber disappear, nuclear membrane reappears. Telophase में nuclear membrane reappears, nucleus ER Golgi body reappears. Telophase की. Mitotic poison. The substance which suppresses halves the division. Azide, cyanide, mustard gas, colchicine. Checks the separation by stopping the polymerization of spindle tubules. Allow polyploidy. Colchicine allow ploiploidy. Even number of chromosome increases. Okay, mitotic poison in the chromosome increases. Even number of chromosome increases. Polyploidy. Number of chromosome increases. Polyploidy. Number of chromosome increases. Polyploidy. Number of chromosome increases. Okay, colchicine checks the separation by checking, stopping the polymerization of spindle microtubules, allow polyploidy. Okay, colchicine, natural mitotic poison obtained from colchicum liliaceae. 2x 4x, polyploidy, number of chromosome increases. Okay, mitotic poison, the substance which suppresses half the division, azide cyanide mustard gas, agglutination, chromosomes grouping. Colchicine checks the separation by checking, stopping the polymerization of spindle microtubule, allow polyploidy. Significance of mitosis: the growth and development of organisms. Childhood, multicellular organization, maintains nucleocytoplasmic ratio, also maintaining the proportionate component of both, maintains the surface area to volume ratio. Cell repair and replacement, maintains the chromosomal number, maintaining the identity. Okay, help in repair and maintenance. Significance of mitosis, right? Equational division is restricted to diploid cell only. Lower plants and social animal haploid cell also divide by mitosis. The result in mitosis, diploid daughter cell are produced. Identical genetic complement. Oh. Diploid daughter cells are produced. Identical genetic complement. Identical genetic components. Diploid daughter cell are produced. Identical genetic complements. Growth of multicellular organism is due to mitosis. Cell growth disturbs nucleocytoplasmic ratio. Cell divides to restore nucleocytoplasmic ratio. The contribution of mitosis: cell repair, cell replacement constantly. Upper layer of epidermis, blood cell, cell of the lining of the gut. One division equals to 20 minutes. Okay, total time upon one division time. Total time. Number of generation. Calculate n. Number of time its cell is undergoing division. Okay, calculation of total daughter of cell after time t. If the time for one division is given, the time for one division is 50 minutes. Total number of daughter produced after 105 minutes. Total time upon division of one cell 105 upon 15, that is 7. Number of generation. Okay, total number of daughter cell 2n. ये तो मुझे कहीं लिखना पड़ेगा. And number of generation. Okay, formula. Number of generation. Number of total generation. N is equals to total time for total cell division. Astaghfirullah, total time. number of generation, total time for total cell division, total time taken for one cell division. Okay, to the power n total number of daughter cells, total number of daughter cells, 2 to the power n, 2 to the power n, total number of daughter cells. आज तक फिर यू विल बी फाइन माँ, मुझे लगता है कि तुम ठीक हो जाओगी. Total number total number of daughter cells to the power n. Okay, what is the total number of generation of cell division? What is the total number of generation cell division is observed during mitosis division? The total daughter cell produced as 64. 64. Okay, तो ये फॉलो करना है. Two twos are 4, 1s are 4, 4 twos are 8, 8 1s are 8, 8 is 16. 16. 2 twos are 4, 4 4s are 16. 2 twos are 4, 4 4s are 4. So 8. 16. 16 twos are 32. 2s are 30, 32, 2s are 64, 64. 1, 2, 3, 4, 5, 6. 2 to the power 6. 6th generation. 6th generation. 2s are 4, 1s are 4, 4s are 8, 8 1s are 8, 8 is 16, 16 1s 16, 17 to 32, 32 1s are 32, 32 2s are 64. Okay, 2 to the power 6. Total number of mitotic division. Total number of mitotic division. Division. Total number of mitotic division is equals to n minus total number of mitotic division x minus 1 x number of daughter cell x minus 1 of daughter cell DAUGH daughter cell. Number of mitotic, total number of mitotic division equals to x minus 1. Number of mitotic division is equals to x minus 1. Total mitotic division x minus 1. 63. X minus 1. Okay, where x is where x is where x is number of total daughter cell. 1 meiosis, 4 pollen grain, 4 megaspore, 1 functional, 1 egg. हो गया। अब कौन सा वाला रिवाइज करूँ? बायोमोलिक्यूल्स। वैसे मुझे बायोमोलिक्यूल्स पढ़ने का दिल नहीं करता, क्या होता है, पढ़ना पड़ता है। जल्दी जल्दी एक बार रिवाइज कर देती हूँ, फटाफट 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 ऐसे करके, जल्दी 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 जल्दी, छेती छेती छेती, स्टॉप इट। बायोलॉजिकल क्लासिफिकेशन ये थोड़ी दिन पहले किया था टेस्ट करते वक्त, एनाटॉमी ऑफ, हाँ कर लेंगे, ये भी करना है। ये चैप्टर मुझे सबसे ज़्यादा गंदा लगता है, लेकिन क्या कर सकते हैं। ओके, फोटोसिंथेसिस इन हायर प्लांट्स, उसको कंप्लीट करना है, पूरा नहीं हुआ है ना। मुझे भी नहीं पता, मैं कंफ्यूज हो रही हूँ। 1 मिनट, हाँ मैं इसको ना कौन सा वाला करूँ देखती हूँ। सारा ही इम्पोर्टेंट लग रहा है अभी करने से, तो बायोमोलिक्यूल से कर लेती हूँ। ओके, अभी बायोमोलिक्यूल्स उसके बाद कर लेंगे हम प्लांट किंगडम। 1 2 3 4 5 आज किया तो 6 बार हो जाएगा। एवोल्यूशन भी करना है। न्यूरल कंट्रोल कोऑर्डिनेशन पूरा नहीं किया है, अच्छा इसे वापस से करना पड़ेगा। फोटोसिंथेसिस इन हायर प्लांट्स, लिविंग वर्ल्ड, बायोलॉजिकल, बायोमोलिक्यूल से कर लेती हूँ। सभी कुछ इम्पोर्टेंट लगता है देखने जाती हूँ तो। Comparison of element present, a comparison of element. Comparison of element present. Earth crust. Oxygen, silicon, sodium, magnesium, hydrogen, carbon, sulfur, nitrogen. Nitrogen is very little, okay? Here science nahi, okay? Human. Oxygen, carbon, nitrogen, calcium, hydrogen, sulfur, sodium, magnesium, silicon. Oxygen, carbon, nitrogen, sulfur. Oxygen, carbon, nitrogen, calcium, hydrogen, then sulfur. Then we have sodium and magnesium, and finally silicon. The component representation of inorganic constituent in living organism. Inorganic constituent: sodium plus PO4 minus SO4 2 minus water, potassium K plus sodium potassium, calcium, magnesium, sodium potassium calcium magnesium. Sodium calcium, potassium, magnesium, water, NaCl, sodium chloride, CaCO4, calcium carbonate, PO4 3 minus, SO4 2 minus, phosphate, sulfate, sodium chloride, and calcium carbonate. Water, magnesium, N plus, sweeter ion. Okay, glycine, alanine, serine, CH2OH. CH2OH, glycine, alanine, serine. Phospholipids, they are found in cell membrane. Lecithin is an example, okay? The chemical and physical properties of amino acids are, okay, the chemical and physical properties of amino acid are essentially of amino, carboxyl and R group. Based on the number of amino and carboxyl group, they are acidic, glutamic acid, basic, lysine, and neutral valine amino acid. Okay, acidic, glutamic acid, lysine, basic, and valine, neutral. Okay, amino acid. Similarly, there are aromatic amino acid. Tyrosine, phenylalanine, and tryptophan. Tyrosine, phenylalanine, and tryptophan. मेरा चार्ज कितना है? It's 23. तो मैं इसको रिमूव ही कर देती हूँ, मुझे साउंड अच्छा नहीं लग रहा है, टूडू कर रहा है बार बार. यहाँ पर ना एक इंसान है ना, उसने बोला है कि समथिंग, छोड़ दो उसका नाम, इम्पोर्टेंट नहीं, बस गाली की तैयारी करनी है क्या, ऐसा बोला है ना। मेरा बहुत ज़्यादा दिल कर रहा है कि हाँ स्टार्टिंग एक्सपेरिमेंट तुझे दे के करूँ, ऐसा बोलने का दिल कर रहा है ना, लेकिन इग्नोर करने में ही भलाई है ना। मेरे बदले में जाके कोई उसको गाली दे देना, ओके, क्योंकि मैं नहीं दे पाऊंगी ना। Neutral valence amino acid. Similarly, there are aromatic amino acid tyrosine, phenylalanine, tryptophan. There are aromatic amino acid tyrosine, phenylalanine, and tryptophan. A particular property of amino acid is the ionizable nature of NH2 and COOH group. Hence, in the solution of different pH, the structure of amino acid changes. B is called deuterion. Deuterion positive charge pani cha negative charge pani cha, both charges cha tara neutral bari, okay. Deuterion, deuterion. Basic lysine and arginine. Neutral. Valine. Tyrosine, phenylalanine. Tyrosine, phenylalanine, and tryptophan. Tyrosine, phenylalanine, and tryptophan. Lipids are generally water insoluble, okay. They could be simple fatty acid, a fatty acid which has a carboxyl group attached to an R group. Fatty acid, a carboxyl group attached to R group. R group could be methyl CH3 or ethyl C2H5 or a higher number of CH2 group, 1 to 19 carbon. For example, palmitic acid has 16 carbon including carboxyl carbon, 16 carbons. Palmitic acid 16 carbon, palmitic acid 20 carbon, arachidonic acid, okay. Saturated without double bond, unsaturated double bond or triple bond. Another simple lipid is glycerol, which is trihydroxypropane, glycerol, trihydroxy propane. CH2 OH glycerol, okay. Lecithin is an example of phospholipid. Lecithin is an example. Lecithin is an example. Lecithin is an example of phospholipid. जोर जोर से खांसने का दिल कर रहा है. Pigments की कैसा बना था? Carotenoid and anthocyanin. Pigment, carotenoid and anthocyanin. Alkaloid, morphine and codeine, alkaloid. Terpenoids, monoterpenes, diterpenes, etc. Excuse me. Carotenoid and anthocyanin. Pigments, alkaloid, morphine and codeine. Terpenoids, monoterpene, diterpene, etc. Okay. Essential oil, lemongrass oil, toxin, abrine and ricin. Lemongrass oil, essential oil, lemongrass oil, toxin, abrine and ricin. Lecithin, concanavalin A. Lectins, concanavalin A. Drugs, vinblastine and curcumin. Polymeric substances, rubber gums, cellulose. Mm-hmm. Pigment, carotenoids, anthocyanin, etc. Pigments, carotenoid and anthocyanin, alkaloid, morphine and codeine. Alkaloid, morphine and codeine. Terpenoids, monoterpene and diterpene. Terpenoids, monoterpene and diterpenes. Essential oil, lemongrass oil. Monoterpene and alkaloid, morphine, codeine. Toxin, aberrin and ricin. Lectins, concanavalin A. Lectins, concanavalin A. Drug, vinblastine and curcumin. Vinblastine and curcumin. Polymeric substances, rubber, gums and cellulose. Okay, these are primary metabolites. Okay, what are, if one wrote if one were to make a list of biomolecules, such a list would have thousands of organic compounds, including amino acids, sugar, etc. For reasons that are given in section 9.10, we can call all the molecules as metabolite. We can call these biomolecules as metabolites. In animal tissue, one noticed the presence of all such categories of compounds shown in figure 9.1. These are called primary metabolites. However, when one analyzes plant, fungal, and microbial cell, one would see thousands of compounds other than these primary metabolites: alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scent, gums, spices. Alkaloids, flavonoids, rubber, essential oils, antibiotics. Okay, alkaloid, flavonoids, rubber, essential oils, antibiotics, even antibiotics. Okay, colored pigment and scents. Colored pigment, scents, gums, spices, scents, colored pigments and gums. These are secondary metabolites. While primary metabolites have identifiable function and play a known role in normal physiological processes, we do not at the moment understand the role or function of all the secondary metabolites in host organisms. However, many of them are useful to human welfare: rubber, drugs, spices, scents, and pigments. Rubber, drugs, spices, scents, and pigments. Okay. Some secondary metabolites have ecological importance. Acid soluble pool. They have molecular weight. Biomacromolecules. Insoluble fraction. Biomacromolecules. Okay. There is one feature common to all those compounds found in acid soluble pool. They have molecular weight ranging from 18 to 800 Dalton approximately. Okay. Not strictly macromolecules. Insoluble fraction. Macromolecules are biomacromolecules. Do not exceed lipids do not exceed 800 Dalton weight. Molecular weight of 800 Dalton. Cl3CCOH, that is trichloroacetic acid. Cl3CCOOH. CCl3COOH. Okay. Water 70 to 90%, protein 10 to 15%, carbohydrate 3, lipids 2, nucleic acid 5 to 7%, ions 1. Okay. Water H2O 70 to 90%, 70 to 90%, right? 70 to 90%. Protein 15 to 10%, 15 to 10%, ions 1%, nucleic acid 5 to 7%, 5 to 7%, 5 to 7% है ना, 5 to 7% है तो. Carbs, fats and lipids. Carbs 3%, fats नहीं, lipids 2%, या फिर carbs 2%, नहीं, carbs 3%, lipid 2%, lipid 2%, lipid 2%, lipid 2%, carb 3%. Okay. Collagen, intercellular ground substances. Collagen, intercellular ground substances. Trypsin, enzyme. Insulin, hormone. Insulin is a hormone. Trypsin is enzyme. Trypsin, enzyme. Insulin, hormone. Antibody. Fight against diseases. Fight infectious agent. Receptors. Receptors of smells and taste. Sensory reception. Smell, taste, hormone. Smell, taste, and hormone. GLUT4, animal glucose transport into cell. Glucose transport protein. GLUT4, animal glucose transport into cell. Animals glucose transport into cell. GLUT4. Okay. Animals glucose transport in the cell. collagen, C-O-L-L-A-G-N, collagen, most abundant in animal world. Rubisco in whole biosphere. Collagen की protein, most abundant protein in animal world. Ribulose bisphosphate carboxylase oxygenase rubisco is the most abundant protein in the whole biosphere. Rubisco protein हो जो biosphere में नहीं most abundant छ, whereas collagen चाहिँ animal world में famous छ या फिर ज़्यादा छ, abundant छ, more छ, amount में ज़्यादा छ. Okay, collagen is the most abundant protein in animal world. Ribulose bisphosphate carboxylase rubisco is the most abundant protein in the whole biosphere. Insulin is a polymer of fructose. Cellulose does not contain complex helices and hence cannot hold I2. Insulin is a polymer of fructose. Insulin is a polymer of fructose. अस्तगफिरुल्लाह, ये नहीं हो सकता मुझसे. दोबारा है ना, 3 बार में आई, ये ज़्यादा हो गया कुछ. Insulin is a polymer of fructose. Insulin, diagrammatic representation of a portion of glycogen. Insulin is a polymer of fructose. Insulin is a polymer of fructose in a polysaccharide chain. Insulin polymer of fructose. Insulin है ना, inulin हो. Inulin polymer of fructose. Inulin polysaccharide हो, is a polymer of fructose. Inulin is a polymer of fructose. The acid insoluble palate also has polysaccharide carbohydrate as another class of macromolecules. Polysaccharides are long chain of sugar. They are thread, literally a cotton thread containing different monosaccharide as building blocks. For example, cellulose is a polymeric polysaccharide consisting of only one type of monosaccharide, glucose. For example, cellulose is polymeric polysaccharide consisting of one type of monosaccharide, that is glucose. Cellulose is a homopolymer. Starch is a variant of this but present as a storehouse of energy in plant tissue. Animals have another variant called glycogen. Inulin is a polymer of fructose. In a polysaccharide chain, say glycogen, the right end is called reducing end and the left end is called non-reducing end. It has branches as shown in the form of cartoon. Such form helical secondary structure. In fact, a starch can hold I2 molecule in the helices portion. Starch I2 is blue in color. Cellulose does not contain complex helices and hence cannot hold I2. Cellulose में helices है उन्हें ना. Plant cell walls are made of cellulose. Plant cell walls are made of cellulose. Paper made from plant pulp and cotton fiber is cellulosic. There are more complex polysaccharides in nature. They have as building blocks amino, sugar and chemically modified sugar. Glucosamine and acetyl galactosamine. Etc. Polysaccharide or chitin. Chitin is a polysaccharide. The complex polysaccharides are mostly homopolymers. मैंने 2 बार कर दिया, इसको कंप्लीट करना ही होगा. Heteropolymer structure of protein. Heteromolecular, the sequence of amino acid, that is positional information of protein. The sequence of amino acid, that is positional information in a protein, which is the first amino acid, which is the second, and so on, is called primary structure of a protein. A protein is imagined as a line. The left end represents by the first amino acid and the right end represents the last amino acid. Left first amino acid and right end represented by last amino acid. The first amino acid is called N-terminal and the last amino acid is called C-terminal. अब मैंने गलत बोला. The first amino acid is called N-terminal. First amino acid N-terminal, left end, okay. Last amino acid C-terminal, right end. Last amino acid C-terminal, right end. First amino acid लेफ्ट एंड. पता नहीं क्यों समझ में नहीं आ रहा है. देखो, जो लेफ्ट टर्मिनल होता है उसे हम लोग फर्स्ट भी बोल सकते हैं और उसे एंड टर्मिनल बोलते हैं. जो सेकंड होता है उसे राइट टर्मिनल बोल सकते हैं और सी टर्मिनल भी बोल सकते हैं. Other regions of the protein thread are folded into other forms. What is called secondary structure? Primary, secondary. Alpha helix, intrapolypeptide H2 bonding, hydrogen bonding. Intrapolypeptide, alpha helix में intrapolypeptide hydrogen bonding. Secondary माने beta pleated पनि हुन्छ नि त, beta pleated shit. Intra or inter polypeptide hydrogen bonding. Three-dimensional view of protein. Tertiary is the absolute necessary for many biological activities of protein. Tertiary structure is absolutely necessary for the many biological activities of protein. Subunit, okay. The manner in which these individual folded polypeptides or subunits are arranged with respect to each other. Linear string of a sphere arranged one upon each other in the form of a cube or plate, etc. in the architecture of a protein, otherwise called the quaternary structure of the protein. Adult human hemoglobin consists of 4 subunits. Two of these are identical to each other, the 2 of alpha type and the 2 subunit of beta type. Human hemoglobin, okay. HB, enzyme, tertiary structure. Inorganic catalysts work efficiently at high temperature. Enzyme get damaged at high temperature, say above 40 degrees. Away from organisms who normally live in extremely high temperature are stable and retain their catalytic power even at high temperature up to 80 to 90 degrees Celsius. Okay, high catalytic power even at high temperature. Thermophilic organism द्वारा enzyme निकाल्न सक्छ. ओके, thermophilic organism बाट enzyme निकाल्न सक्छ. Chemical reaction. Rate of or chemical process refers to the amount of product per unit time. Rate del P upon del T. Rate of physical or chemical process refers to the amount of product formed per unit. Time. Rate can also be called as velocity if the direction is specified. A general rule of thumb is that rate doubles or decrease by half for every 10 degree change in either direction. Doubles or decrease by half for every 10 degree change in either direction. Catalyzed reaction proceed at rates vastly higher than that of uncatalyzed one. Catalyzed reactions proceed at rates vastly higher than that of uncatalyzed ones. When enzyme catalyzed reaction are observed, the rate would be vastly higher than the same but uncatalyzed reaction case. Rate can also be called velocity if the direction is specified. Rate of physical and chemical process are influenced by temperature among the factor. A general rule of thumb is that rate doubles or decrease by half for every 10 degree degree centigrade change in either direction. Catalyzed reaction proceed at rate vastly higher than that of uncatalyzed one. When enzymes catalyze a reaction are observed, the rate would be vastly higher than that of uncatalyzed reaction. For example, carbon dioxide plus water in presence of carbonic anhydrase gives carbonic water, carbonic acid, H2CO3, carbonic acid, H2CO3. Carbonic acid. In the absence of enzyme, this reaction is very slow, with about 200 molecules of H2CO3 being formed, carbonic acid being formed in an hour, 200 molecules in an hour. However, by using enzyme present in the cytoplasm called carbonic anhydrase, the reaction speeds dramatically with about 6 lakh molecules being formed every second. 6 lakh molecule. Okay, 1 hour 200 molecules. 6 lakh molecule formed every second. The enzyme has accelerated the reaction rate about 10 million times. Thousands of types of enzymes, each catalyzing a unichemical or metabolic reaction, multi-step chemical reaction. Different enzymes. Metabolic pathway. A multi-step chemical reaction when each of the step is catalyzed by the same enzyme complex or different enzymes is called metabolic pathway. For example, glucose to pyruvic acid. 10 different enzymes. Catalyzed metabolic reaction. 10 different enzymes catalyze metabolic reaction. In yeast, during fermentation, the same pathway leads to production of ethanol, alcohol. Hence, in different conditions, different products are possible. Potential energy versus progress of reaction. Concept of activation energy. Okay, the energy level difference between S and P, substrate and product. If product is at lower level than substrate, the reaction is exothermic. The difference is average energy content of S from that of this transition state is called the difference in average energy content of substrate from that of this transition state is called activation energy. Factors affecting enzyme, the temperature, concentration of substrate. pH percent, so concentration of substrate, Vmax. Km, Berzelius constant, Vmax by 2. Which is not okay. The reaction ultimately reaches the maximum velocity Vmax, which is not exceeded by any further rising concentration of the substrate. Competitive inhibitor. Inhibitor closely. Hey, when the binding of the the substrate cannot bind. As a result, the enzyme acts. Inhibition of succinic dehydrogenase by malonate. Inhibition of succinic dehydrogenase. Inhibition of succinic dehydrogenase. Inhibition of succinic dehydrogenase. I think वो थोड़ा ज़्यादा हो गया. Inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate. Succinate. 3. Not 3. 60, 70, 80. 2 hours. Example, inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate. Which closely resembles the substrate controls by bacterial pathogen. Succinate. Such competitiveness are often used in the control of bacterial pathogen. Used in the control of bacterial pathogen. Used in the control of bacterial pathogen. Okay. Cofactors. Enzymes are composed of one or several polypeptide chains. Multiple वो मैंने लगता है मैंने ना इम्पोर्टेंट पेपर ही फेंक दिया. ओ नहीं नहीं यहाँ पे नहीं है. नहीं है. मुझे लगता है कि आज सुबह या फिर कब मिला था मैंने फेंक दिया क्या. आई थिंक मैंने इसके अंदर पैसा भी डाल के रखा है. नहीं है शायद फेंक दिया. भाई साहब को इतना इम्पोर्टेंट पेपर कैसे फेंक सकते हैं. Types of enzyme. Simple enzyme, only protein present in active form. Only protein. Simple enzyme. Simple enzyme, only protein present in active form. Conjugate enzyme. Conjugate enzyme. Holoenzyme. Okay, holoenzyme. Inactive protein. Opoenzyme. Non-protein cofactor and opoenzyme. Holoenzyme cofactor plus opoenzyme. Holoenzyme. Cofactors. Enzymes are composed of several polypeptide chains. However, there are a number of cases in which non-protein constituent cofactor, non-protein constituent constituent called cofactors are bound to the enzyme to make the enzyme catalytically active. In this instances, the protein portion of the enzyme is called opoenzyme. Three kinds of cofactors may be identical: prosthetic groups, coenzymes, and metallions. Enzymes. MES enzymes, okay. However, there are a number of cases in which non-protein constituent called cofactors are bound. Non-protein cofactors, okay. Enzyme to make enzyme catalytically active. In this instance, the protein portion of enzyme is called opoenzyme. Opoenzyme plus cofactor. मैंने क्या किया यार, मैं खुद को ना मार दूं. मैंने कहीं रख दिया है, जो हमेशा मैं नोट ना खो जाती हूँ, खो देती हूँ, पता नहीं क्या एग्जैक्टली क्या बोलते हैं मुझे नहीं पता, लेकिन ना खो जाता है मेरा नोट्स। इतना सा पेपर में ही तो लिखी थी, ये नहीं है। Prosthetic coenzymes, okay. Enzymes. Simple enzyme and simple enzyme. Holoenzyme, okay. Holoenzyme, optoenzyme, inactive protein plus holoenzyme. Optoenzyme plus cofactor. Non-protein, okay. Optoenzyme and cofactor. Optoenzyme and cofactor. Optoenzyme inactive protein, okay. Cofactors non-protein. Cofactors, okay. The protein portion of enzyme is called optoenzyme. Prosthetic coenzyme and prosthetic metal ion, prosthetic group coenzymes, three types of cofactors: prosthetic group coenzyme and metal ion. Prosthetic group coenzyme 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 metal ion. inorganic cofactors, coenzymes and prosthetic group. Holoenzyme, apoenzyme plus cofactor, apoenzyme plus cofactor, apoenzymes plus cofactor. Okay. Prosthetic group, organic compound, and are distinguished from other cofactors in that they are tightly bound to the apoenzyme. Peroxidase and catalase, which catalyze the breakdown of hydrogen peroxide. Catalyze the breakdown of hydrogen peroxide. Catalyze the breakdown of hydrogen peroxide. Peroxidase and hydrogenase. I don't know. Peroxidase and catalase. 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 Hydrogen peroxide to water and oxygen. Heme is prosthetic group, and it is part of active site of enzyme. Part of prosthetic group, and it is part of active site of enzyme. Coenzymes are also organic compounds, but their association with apoenzyme is also transient, usually occurring during the course of catalysis. Vitamins. But the association with the apoenzyme is also transient, usually occurring during the course of catalysis. Number of different enzyme catalyzed reactions. The essential chemical components of the coenzymes are vitamins. Example. Okay. वो ना सेकंड स्लॉट कंप्लीट हो गया। ओके, टाइमर बच गया। हमने इस बार भी थोड़ा ज़्यादा पढ़ा क्योंकि बीच में मैं टाइमर पॉज करके स्टार्ट करना भूल गई थी, और ये 60 मिनट का नहीं 70 मिनट का सेशन था। ये इसको ढूंढ रही थी, यहाँ पर था। Isomerase, ligase, oxidoreductase, dehydrogenase, transferase, isomerase, ligase, lyases, isomerase, hydrolase, transferase, oxidoreductase, dehydrogenase, oxidoreductase, dehydrogenase, transferase, isomerase, lyases, ligase, transferase, oxidoreductase dehydrogenase enzyme which catalyze oxidoreduction between two substrate. Okay, enzyme which catalyze oxidoreduction between two substrate. Yes, oxidize. Transferase, enzyme catalyzing a transfer of group G other than hydrogen between a pair of substrate as an ester. Transferase. Hydrolase, enzyme catalyzing hydrolysis of ester ether peptide glycosidic CC carbon carbon and C halide alkyl halide or PN bonds. Hydrolase, enzyme hydrolysis of ester peptide ether glycosidic CC C halide and PN bonds. Lysis, enzyme that catalyzes removal of group from substrate by mechanism other than hydrolysis. Leaving double bond, okay. Enzyme that catalyze removal of group from substrate by mechanism other than hydrolysis. Leaving double bond, okay. Isomers include all enzyme catalyzing interconversion of optical, geometrical or positional isomers. Conversion of optical, geometrical or positional isomers. Ligases, enzyme catalyzing the linking together of two compounds. Example, enzymes which catalyzes joining of carbon, oxygen, carbon, sulfur, carbon, nitrogen, PO, etc. 10 मिनट का ब्रेक लेना है ना, 12 मिनट का हो गया, ये लो 10 मिनट का ब्रेक। ये लोग कितना, मैं इतना बोल रही हूँ कि ऑर्गेनाइज रखो, रख रहे हैं, क्या ही कर सकते हैं। Hydrolysis of bond. मैं चाय बना के आती हूँ, ओके। 10 सेकेंड ही है। आ गई वापस, खाना भी खाया और चाय भी बनाया। कितने मिनट का सेशन रखूं? बोलो, चैट सेक्शन ऑन है अभी, लेकिन अब ऑफ करूंगी इसके बाद। 90 का कौन कौन बोलता है इसके साथ 90 मिनट का पढ़ने के लिए? शुभम के साथ साथ सच्ची में 90 मिनट का सेशन रखना है क्या? 90? हाँ, तुम तो ऑब्वियसली पढ़ोगे ही, तुम ही ने बोला है तो तुम तो ऑब्वियसली पढ़ोगे ही। श्री, आर यू देयर? 90 मिनट का बोल दिया इसने। इसको ना मुझे मारने का दिल कर रहा है, लेकिन मैं मार नहीं सकती क्योंकि ये एक जगह नहीं बैठा है। फ्लाई है, फ्लाई। तीन सौ 10 इज बैक, आई कैन सी। 100 मिनट का पक्का। मैंने बेकार में चिल्ली खाया ना, अब मुझे पेन करने लगा फिर से। मेरी बॉडी स्पाइस डाइजेस्ट या फिर टॉलरेट कर ही नहीं सकती, फिर भी मैं खाती रहती हूँ। मुझे लगा कि मैं इस बार इसको मार दूंगी, लेकिन मैं नहीं मार पाई। ऊपर उड़ गया, नहीं मार पाई। वैसे मैंने कर दिया है 100 मिनट का, लेकिन आई एम प्रीटी श्योर कि 100 मिनट कंप्लीट करने के बाद हम लोग फिर पढ़ने लायक नहीं बचेंगे, क्योंकि हम लोगों को बहुत बुरा लगेगा इतना पढ़ने के बाद। मैं इसको मार के पढ़ूँ कि ऐसे ही पढ़ूँ? एक मिनट, हाँ मैं इसको एक बार मार के पढ़ देती हूँ, फिर ये डिस्टर्ब करने आ जाएगा ना, डिस्ट्रैक्ट करेगा। मैं इसको मार ही नहीं पाऊंगी यार। आई थिंक इस बार मैंने मारा, लेकिन कहाँ गिर गया वो नहीं पता। आई थिंक इस बार मैंने उसको मार दिया जिस तरह मुझे, क्योंकि उड़ भी नहीं रहा है ना। डेड बॉडी मिली ना कल साफ करते वक्त, मैं दिखाऊंगी। मैंने तो सेशन के लिए एक गुड लक बोला, मुझे लग रहा है कि आपने मुझे ना एग्ज़ाम के लिए गुड लक बोल दिया। मैंने तो सेशन के लिए ही बोला था सिर्फ। अर्जुन, अर्जुन राइट, अर्जुन मैं सिक्किम से हूँ। अगर तुम्हें नहीं पता सिक्किम कहाँ है, तो सिक्किम इंडिया में ही है और नॉर्थ ईस्ट साइड में है। ओके, ये लो मैंने कमेंट ऑफ भी कर दिया। SIKK आई एम सिक्किम, तुमने 2 बार लिखा ना क्वेश्चन, तो मैंने भी 2 बार आंसर दे दिया। हाँ मैं स्टार्ट कर देती हूँ। ओके, सब लोग पढ़ने के लिए बैठ जाओ। ये लो ऑफ कर दिया। स्टार्ट कर रही हूँ. ओके, गुड लक एवरीवन. A possible location where this could be happening. Okay, PS1 is functional. The electron is circulated within the photosystem and the phosphorylation occurs due to cyclic flow of electron. A possible location where this could be happening is in the stroma lamella, while the membrane or lamella of the grana have both PS1 and PS2. Is in the stroma lamella. Cyclic flow of electrons, stroma lamella, stroma lamella, while the membrane or the lamella of the grana have both PS1 and PS2. Stroma lamella ma cyclic phosphorylation huncha, non-cyclic phosphorylation grana ma huncha. Okay, while the membrane or the lamella of the grana have both PS1 and PS2. The stroma lamella membrane lacks PS2 and like PS2 as well as NADP reductase enzyme. NADP reductase enzyme. The excited electron does not pass on to the NADP plus, but the excited electron does not pass on to the NADP plus, but is cycled back to the PS1 complex through the electron transport chain. The cyclic flow hence result on the synthesis of ATP but not of NADPH plus H plus NADPH plus H plus. Wavelength beyond 680 nanometer are available for excitation. For excitation उससे पहले ना 11th का ही किया था. हाँ, cell cycle and cell division. Electron acceptor photosystem 1. Okay, light ADP plus IP ATP. Electron transport system chlorophyll P700 photosystem 1 electron acceptor. Location cyclic phosphorylation location का हुन्छ स्ट्रोमा ल्यामेलामा हुन्छ. के स्ट्रोमा ल्यामेलामा हुन्छ. कहाँनिर हुन्छ स्ट्रोमा ल्यामेलामा. P700 for chlorophyll PS photosystem 1 P700 photosystem 1 photosystem 2 680 nanometer. Okay, P680 light LHC light harvesting complex electron acceptor. Okay, antenna. Then electron transport system Z scheme or light reaction. Z scheme of light reaction. Cyclic phosphorylation. Z scheme of light reaction. Okay, electron transport system ETS ADP1 र phosphate बाट हुन्छ ATP1 हुन्छ. के यसलाई phosphorylation पनि भन्छ. photophosphorylation light को थ्रु बाट भन्ने भने है. यही त था. एउटा एक्सन स्पेक्ट्रम हुन्छ, एउटा अब्जर्प्सन स्पेक्ट्रम हुन्छ. ब्लू र रेड रिजनमा हाई हुन्छ. कहीं तो था लेकिन मैं भूल गई, पता नहीं कहाँ था. Electrons available due to splitting of water. Electron के गर्छ प्रोवाइड गर्छ. इलेक्ट्रोन प्रोवाइड गर्छ. के बार फोटो सिस्टममा कसले इलेक्ट्रोन दिन्छ. फोटो सिस्टम 2 मा इलेक्ट्रोन वाटर स्प्लिटिङ बाट गेन गर्छ. Okay, electrons available due to splitting of water. Okay, splitting of water is associated with PS2. Okay, Z scheme भन्छ. जोसेफ प्रेस्ले. ओके, बेल जार एक्सपेरिमेन्ट. जोसेफ प्रेस्ले को बादमा आयो जन आइगनहाउस डाइकेसिस. जन आइगनहाउसले के बतायो भन त. सनलाइट इज एसेन्शियल फर फोटोसिन्थेसिस. सनलाइट इज एसेन्शियल. aquatic plant मा गएको थियो. ब्राइट सनलाइट, स्मल बबल्स, डार्क दे डिड नट. ओके, ग्रीन पार्ट्स while in the dark they did not. Okay, only green part of plant could release oxygen. 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 जोसेफ प्रिस्ली बेल जार एक्सपेरिमेंट, उसके बाद में आइगन हाउस आया, जेन आइगन हाउस, सनलाइट इज एसेंशियल. He experimented in aquatic plant, bright sunlight, small bubbles. Bright sunlight हुँदाखेरि small bubbles देखियो. Green parts, okay, where small bubbles were formed around the green parts, while in the dark they did not. Okay, later he identified these bubbles to be of oxygen. Only the green part of the plant could release oxygen. Green part of the plant could release oxygen. Okay, त्यहाँदेखि जुलियस वन सैक आयो. जुलियस वन सैक. Glucose is usually stored as starch. Glucose is usually stored as starch. Glucose starch को फर्म में स्टोर हुन्छ भनेर जुलियस वन सैकले बतायो. कसले बतायो? जुलियस वन सैक. जुलियस वन सैक. Glucose is stored as starch. Is located in spatial bodies, later called chloroplast, within plant cells. Okay, he found that the green parts in the plant is where glucose is made, and the glucose is usually stored as starch. Glucose usually के हुन्छ starch को फर्ममा स्टोर भएको हुन्छ. I think I have to give the test. देखते हैं. ओके, केमिस्ट्री और फिजिक्स दोनों में से एक का है तो मैं नहीं करूँगी. पता ही नहीं है क्या करना है. बायो का है तो हल गया. केमिस्ट्री क्लास 12 पार्ट 1 है. पार्ट 1 ना पार्ट 2 दोनों ही नहीं किया है, तो आई थिंक यहाँ से तो नहीं दे सकते. तो एटलीस्ट मिनिमम 50 तो होना चाहिए ना, लेकिन 50 भी नहीं है. पता नहीं कैसे करेंगे. 50 हुआ तो ही 360. Julius von Sack provide evidence for production of glucose when plants grow. Glucose is usually stored as starch. Glucose is usually stored as starch. His later studies showed that the green substances in plant. Okay. His later studies showed that the green substances in plant chlorophyll, as we know it now, is located in spatial bodies, later called chloroplast, within plant cell. Okay. Green parts in plant where glucose is made, and the glucose is usually stored as starch. Okay, Julius von Sackले भन्यो ग्लुकोज को फर्ममा ग्लुकोज चाहिँ स्टार्च को फर्ममा स्टोर भएर बसेको हुन्छ कहाँनिर? क्लोरोप्लास्टमा. ए क्लोरोफिलको कुरा गर्दैछ. क्लोरोफिल कहाँनिर हुन्छ? क्लोरोप्लास्टमा. ओके, TW एंगलमैन फर्स्ट प्रिस्टले, देन जेन आइगनहाउस, जुलियस वन सैक र TW एंगलमैन. TW एंगलमैनले प्रिज्ममा के गर्यो? लाइट स्प्लिट गर्यो. क्लाडोफोरा के क्लाडोफोरा प्लेस्ड इन सस्पेंशन अफ एरोबिक बैक्टेरिया युज गरेको थियो. क्लाडोफोरा एल्गियो, क्लाडोफोरा र बैक्टेरिया, एरोबिक बैक्टेरिया. ओटो एभोल्युसन ब्लू एंड रेड रिजनमा के थियो? ज्यादा थियो. It resembles roughly the absorption spectrum of chlorophyll. EP. Cornelius van Niel, purple and green photosynthetic bacteria. Purple and green bacteria demonstrated the photosynthesis is essentially a light-dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide and पता नहीं क्या हो रहा है, carbohydrates. Okay, the green part, water. In green plants, water is the hydrogen donor and oxidized to O2 as present state. When H2S instead is the hydrogen donor for purple and green sulfur bacteria, the oxidation product is sulfur or sulfate depending on the organisms and not O2. Hence, he inferred that O2 evolved by the green plant. O2 evolved by green plants come from water. O2 evolved. Oxygen evolved by green plants. Oxygen evolved by the green plant comes from H2O, not from carbon dioxide. Julius Von Zach, T.W. Engelmann. T.W. Engelmann, using a prism. Cladofera placed in a suspension. Aerobic bacteria, Biocornellus, and purple and green bacteria demonstrated that photosynthesis is essentially a light-dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrate. This can be expressed by: in green plant, water is the hydrogen donor and is oxidized to O2. Some organisms do not release O2 during photosynthesis when H2S instead is the hydrogen donor. For purple and green sulfur bacteria, the oxidation product is sulfur or sulfate depending on the organism and not O2. Hence, he inferred that the O2 evolved by the green plants comes from water. O2 evolved by green plants comes from H2O, not from carbon dioxide. The O2 released in the water. Okay, here's 6H12. O2 released in the form of water. Okay. ATP and ADPH. क्या था इसका? Product of light reaction ATP and NADPH. अलॉन्ग विथ क्या था? ATP NADPH. और क्या चाहिए लाइट रिएक्शन के लिए या फिर डार्क रिएक्शन के लिए? ATP NADPH चाहिए अलॉन्ग विथ कार्बन डाइऑक्साइड. Hypothesis. Say plant, some O2 binds to rubisco and hence CO2 fixation is decreased. Does not occur. Mesophyll cells lack rubisco. Bundle shaped cell. 1 minute. How? Well, one of the correct use भी नहीं है. 8, 67, 68. 68 go on. Try abicron borer, bico4. Try AC cotton bollworm, enzyme ADA deficiency. डिको2 भाई सही तो लगाया था डिको2 अल्फा 1 एंटीट्रिप्सिन Axoneme, cilia and flagella, cartwheel pattern, centriole, chromosome and mitochondria, crista, mitochondria, satellite, chromosome. 2%. The flippers of penguin and dolphins are the example of flippers of penguin and dolphin. Assertion. Breastfeeding during initial period of infant growth is recommended by doctors for bringing a healthy baby. Colostrum contains several antibodies absolutely essential to develop resistance for newborn baby. Which of the following is not a component of fallopian tube, uterine fundus? Which of the following statement is incorrect? Bioreactors are used to produce pons. Hypothalamus, medulla and cerebellum provide additional space for neuron regulated posture and balance control. Balance, control respiration and gastric secretion, connects different region of brain, neurosecretory cell, hypothalamus, neurosecretory cell, cerebellum, region of the brain, connects different region of the brain, control respiration and gastric, medulla oblongata को 2, CO2. Dandaki, space for neurons, provide additional space for neuron, regulate posture and balance. Connect respiration, controls respiration and gastric skin, connects different region of the brain, cerebellum. Hypothalamus को 4, B को 4. Pons consist of fiber tract that interconnect different region of brain. Pons connect different region of the brain. Pons consist Pons consist of fiber tract that interconnect different region of brain. Where is that? Pons connects different region of the brain. Pons consist of fiber tract. Pons connect different region of brain. Cerebellum. Cerebellum has a very convoluted surface in order to provide the additional space for many more neurons. Neurosecretory cell को बोल रहे हैं. The medulla of the brain is connected to the spinal cord. The medulla contains center which control respiration, cardiovascular reflexes, and gastric secretion. Okay. Medulla contains centers which control respiration and gastric secretion, but cardiovascular भी तो है. Okay, then medulla controls respiration and gastric secretion along with cardiovascular reflexes. Cerebellum. Cerebellum का क्या दिया है? Provide additional space for neurons, regulate posture and balance. Has a very convoluted surface in order to provide additional space for neurons. Additional space. Cerebellum. ये ना फिर से इसे साउंड करने लगा. Cerebellum. Provide additional space for neurons. Additional space for neurons. Cerebellum. Regulate posture and balance. Regulate posture and balance. Regulate posture and balance, cerebellum. Regulate posture and balance. Cerebellum. Pons connects different region of brain. Cerebellum. Additional space for many more neurons. बस इतना ही दिया है. Medulla controls respiration and gastric secretion along with cardiovascular reflexes. भी medulla ही control कर रहा है. Hypothalamus. Neurosecretory cells. ना 1, 2, 3. D4 वही तो था. D4 ऐसे भी कर सकते थे ना. B को इन कर सकते. B को 3. हाँ ये कर सकते थे वैसे भी. Hypothalamus. Neurosecretory cell. Hypothalamus neurosecretory cell. Hypothalamus neurosecretory cells. Lipases, nuclease, protease and amylase. I think ये बायोटिक का है ना. Lipases, ester bond, nuclease, phosphodiester bond, protease, peptide and amylase, glycosidic bond. Excuse me. Which of the following is not a natural traditional contraceptive method evolved? Diaphragm, cervical cap and walls. Walls are barriers. PYQ. Walls are barrier. Diaphragm, cervical caps and walls are barriers. Diaphragm, cervical and walls. Charging only. Lactational amenorrhea, coitus interruptus, withdrawal or coitus interruptus, periodic abstinence and periodic abstinence, withdrawal or coitus, lactational amenorrhea. AV bundle, Purkinje fiber, AV node bundle. Following are the stages of pathway conduction of the axon potential through the heart. ऐसे नोड, then हम लोग में ऐसे नोड पर आंसर sinoatrial node. What is answer? AVN, atrioventricular node. Then interventricular septum बोला, interventricular, atrial ventricular septum. Septum. SA node, AV node, okay. AVN, then AV bundle, then bundle branches, Purkinje fiber at the end. Okay, first one कितना था? E आणि EA. SA node, AV node. Then AV bundle, Purkinje fiber. Last में से भी bundle branch. Order, okay. Sun. Action potential, conduction of action potential, okay. Sun. AVN, nodal fiber, atrioventricular bundle, AV bundle, atrioventricular bundle, AV bundle, from AVN and passes through the atrioventricular septa to emerge on top of the interventricular septum and immediately divide into right and left bundle, okay. Bundle branches. The branches give rise to minor fiber, finally Purkinje fiber. PYQ 2024. Then AVN, sinoatrial node, atrioventricular node, AV bundle, then right and left bundle, bundle branches, finally Purkinje fiber, fifth, okay. Sun, then AVN, AV bundle, third, AV bundle, then right and left bundle, finally Purkinje fiber. Pleurobrachia, radula, stomachoid, and ear bladder, okay. Ostrich size, ctenophora, and mollusca. Pleurobrachia, hemichordata, pleurobrachia, hemichordata, eco4. Pleurobrachia. Balano glasses and sacco glasses है वो. Pleurobrachia इसमें नहीं है. Radula cylindrata. Ctenophora दिया है यहाँ पर. अरे मोलस्का में था रेडुला, तो भूल गई है. Ostrich size, stomachoid, pleurobrachia. Ctenophora में था pleurobrachia, okay. Bladder, ostrich size. viral test typhoid. Okay, SC2. Yeah. Dust mites. Okay. Common cold, hemozoin. Malarial parasite, Plasmodium. Rhinovirus. Mollusca redula. Redula mollusca. Mollusk, file-like rasping organ feeding radula. Okay, visceral hump, muscular foot. Example: Pila, apple snail, pink tadpole, pearl oyster, sepia, cuttlefish, loligo squid, octopus devilfish, aplysia, sea hare, dantalium, tusk shell, Catopleura, chitin. Apple snail, Pila, pink tada, pearl oyster, sepia, cuttlefish, loligo squid, octopus devilfish, aplysia, sea hare, and dantalium, tusk shell, Catopleura, chitin. Example of mollusk. Apple snail, Pila, apple snail. Okay, mollusk. Radula, loligo squid, right? Dentalium, tusk shell, tusk shell, octopus, devilfish, Catopleura, chitin, aplysia, sea hare, sepia, cuttlefish. Pink tada, pearl oyster, Pila, apple snail, pink tada, pearl oyster, mollusk example. Okay, mollusca, sepia, cuttlefish, loligo squid, octopus devilfish, aplysia, sea hare, dantalium, tusk shell, Catopleura, chitin. Okay, then we have pleurobrachia, stomachoid, ear bladder, ostrich thighs, myxin, cyclostoma, petromyzone lamprey, and myxin hagfish. Cyclostoma, petromyzone lamprey, myxin hagfish. Okay, cyclostoma, chondrich thighs and ostrich thighs. Cyclostoma jawless. Then we have Piscisma, chondrich thighs, ostrich thighs, bear fins. Okay, chondrich thighs and ostrich thighs, superclass. Bears, Pisces and tetrapoda. Okay, एक ना था. Laxtra class, cyclostoma. Myxin and hagfish, cyclostoma. Cyclostomata. class chondrich thighs and ostrich thighs. Ostrich thighs and chondrich thighs. Okay. Chondrich thighs. Prestes, Sophis, Prestes, Sophis, Prestes, Sophis. Chondrich thighs. Okay. Chondrich thighs. Ostrich thighs. Both cycloid and ctenoid scale. Cycloid and ctenoid scale. Cycloid and ctenoid scale. Ostrich thighs have both cycloid and ctenoid scale. Ostrich thighs. It includes both marine and freshwater fishes with bony skeleton. Ostrich thighs, bony skeleton, both marine and freshwater fishes. Their body is streamlined. Mouth is mostly terminal. Body is streamlined. Mouth is terminal. They have 4 pair of gills. They have 4 pair of gills which are covered by an operculum. Their body is streamlined. Mouth is mostly terminal. They have 4 pair of gills which are covered by operculum. 4 pair of gills हुन्छ. They have 4 pair of gills. Ostrich thighs को मा 4 pair के हुन्छ gills हुन्छ. They have 4 pair of gills which are covered by an operculum. Operculum on each side. Skin is covered by cycloid and ctenoid scale. Cycloid and ctenoid scale. Ear bladder is present which regulate buoyancy. See. Buoyancy. Buoyancy or buoyancy? Buoyancy, okay. Ear bladder. Ostrich thighs. Four pair of gills. 4 pair of gills covered by operculum. Covered by operculum. They have ear bladder to maintain buoyancy. Heart is 2-chambered, 1 auricle, 1 ventricle. They are cold-blooded animals. Sexes are separate. Fertilization is usually external. They are mostly oviparous and development is direct. Exocetus, flying fish. Aquarium, betta fish. Pterophyllum, angelfish. Okay. Freshwater र marine दुईटै हुन्छ. Mouth terminal हुन्छ. Streamlined body हुन्छ. Operculum present हुन्छ. 4 pair gills lit हुन्छ. त्यहाँदेखि what else? Air bladder छ. Buoyancy maintain गर्न सक्छ. आफ्नो regulate गर्न सक्छ. Swim गरिबस्नु पर्दैन भनेपछि. Then sexes different हुन्छ. Separate हुन्छ. Cold blooded animal हो. External fertilization हुन्छ. External fertilization हुन्छ अनि ostrich thighs को भनेकै they are bony, okay. अनि क्या था? Cycloid and ctenoid scales. Scales cycloid and ctenoid हुन्छ. They have 4 pair of gills which are covered by an operculum, okay. On each side, skin is covered by cycloid or ctenoid scale. Cycloid नभए ctenoid scale हुन्छ, okay. Cycloid नभए ctenoid scales. For external oviparous हुन्छ. Development direct. Oviparous and direct development. Direct development हुन्छ. Example, fighting fish, betta fish, pterophyllum, angelfish, pterophyllum, and they have marine र के गरेको छ marine अलगै freshwater अलगै र aquarium मा betta fish fighting fish, pterophylum, angelfish. Okay, betta fish, fighting fish, pterophylum, angelfish. Marine ma hippocampus and exocetus. Hippocampus seahorse, exocetus flying fish. Exocetus flying fish. Freshwater labio rohu. Labio rohu. Clarius magur. Freshwater labio rohu, kotle, and then Clarius mangur. Clarius and magur. Clarius magur. Clarius magur. Clarius magur. Okay, labio rohu. Marine ma exocetus flying fish, hippocampus seahorse, freshwater labio, kotle and clarius. Labio rohu, kotle and clarius magur. Clarius magur. Clarius magur. And clarius is magur. Okay. Cycloid and tenoid scale, ostrich thighs. Cycloid and tenoid scale, ostrich thighs. Okay. Prestige surface. Palacoid scale हुन्छ यिनीहरूको scale ले होला devoid of scale. Cyclostomata devoid of scale हो. Palacoid scale chondrich thighs. Cycloid and tenoid ostrich thighs. Okay, pleurobrachia, ctenophora. Pleurobrachia and ctenoplana are okay. Pleurobrachia and ctenoplana. Pleurobrachia, PYQ. Okay. Pleurobrachia, ctenophores. Pleurobrachia, ctenophores, pleurobrachia. Okay, ctenoplana pani ctenophora ma आउँछ. Pleurobrachia. And finally, stomochord hemichordata. Stomochord hemichordata. Excuse me. Hemichordata. Stomochord. Okay. Stomochord hemichordata. Stomochord. Hemichordata was earlier considered as subphylum under phylum Chordata, but now it is placed as a separate phylum under non-Chordata. Hemichordata have a rudimentary structure in the collar region, trunk, collar, and proboscis. Proboscis, collar, trunk. But now it is placed as a separate phylum under non-Chordata. Hemichordata have a rudimentary structure in collar region called stomochord. ला मलाई पूरा डर लाग्यो, मतलब कि I think मुझे डर लग रहा है. Hemichordata have a rudimentary structure in collar region called stomochord, or structure similar to notochord. Hemichordata have a rudimentary structure in collar region called stomochord, a structure similar to notochord. Notochord. A rudimentary structure in collar region called stomochord, a structure similar to notochord. Proboscis, collar, and trunk. This phylum consists of a small group of worm-like marine animals with organ system level of organization. They are bilaterally symmetrical, triploblastic and coelomate animals. The body is cylindrical and is composed of an anterior proboscis, a collar, and a trunk. Collar, trunk, proboscis. Circulatory system is of open type. Respiration takes place through gills. Excretory organ is proboscis gland. Sexes are separate. Proboscis gland, excretory organ. Respiration takes place through gills. Circulatory system is of open type. Development is indirect. Sexes are separate. Fertilization external. Balanoglossus and saccoglossus. Anterior proboscis. Collar, trunk. Common cold, rhinovirus, hemozoin, viral test. Okay. Annelids are true. Annelids are true coelomate. Annelids. As Calmentis, that's one. Yes, correct. Poriferans are pseudo coelomate. Nah, they are acoelomate. As Calmentis are coelomate. They are pseudo coelomate. False. Platyhelminthes are pseudo coelomate. No, they are again platyhelminthes. Acoelomate they are. यहाँ आगे तक ही दिया होगा ना ये बात. पागल हो जाऊंगी ऐसा लग रहा है, लेकिन कोई बात नहीं. Acoelomate, platyhelminthes. यहाँ पर absent platyhelminthes तक absent है. Okay. As Calmentis pseudocoelomate. As Calmentis pseudocoelomate. Then finally poriferans absent, annelids are coelomate. Annelids coelomate. Excuse me. In both sexes of cockroach, a pair of drones. Cockroach तो नहीं दिया है ना इसमें. फ्रॉग ही दिया है। In both sexes of cockroach, a pair of joint filamentous structure called anal cerci are present on 10th segment. Anal cerci. 26. 6. In both sexes, the 10th segment, in both sexes of cockroach, a pair of joint filamentous structure called anal cerci are present. Anal cerci, 10th segment, okay, 10th segment one. In both sexes, 10th segment is the question answer. 10th segment in both sexes of cockroach, a pair of joint filamentous bears a joint filamentous structure, pair of joint filamentous structure called anal cerci. Anal cerci, this is PYQ. मैं सही करके आई थी, अच्छा है. Statement 1. The presence or absence of a reliable indicator of false. Statement 1 is true, statement 2 is false. Anal cerci, AC. 10th segment में, okay, 10th segment में. Amniocentesis, statutory, ban on amniocentesis, means of female foeticide. Statement based, okay. Constant gene pool. Hardy-Weinberg equilibrium. Okay. इससे पहले ठीक. Common cold. Which one of the following factors will not affect Hardy-Weinberg equilibrium? Gene flow, genetic drift, mutation, genetic recombination, and natural selection. Gene flow, genetic drift, mutation, gene flow, genetic drift, gene flow, genetic drift, genetic drift, mutation, mutation, genetic recombination, recombination, genetic drift, gene flow, genetic drift, mutation, genetic recombination, and natural selection, natural selection. Okay, constant gene pool. क्या हो ये gene pools? नहीं पता यार, पता नहीं क्या है ये gene pool. Gene migration, gene migration. Migration of a section of population of another place and population occurs. Gene frequency change in the original as well as new population. New genes alleles are added to the new population and these are lost from the old population. There would be gene flow if the gene migration, gene flow. There would be gene flow if this gene migration happens multiple times. Gene migration, gene flow, genetic drift, gene flow, genetic drift, gene flow, genetic drift, gene mutation, genetic flow, genetic drift, genetic recombination. Gene flow. एक मिनट, हाँ जीन फ्लो जेनेटिक जीन माइग्रेशन दिखाया, जीन माइग्रेशन तो सही है, जेनेटिक रिकॉम्बिनेशन जेनेटिक ड्रिफ्ट तो दिया भी है. कांस्टेंट जीन पूल. Malaria isn't malaria. Nah, it's hemozoin. Hemozoin and plasmodium. Plasmodium, hemozoin, okay? Hemozoin. Then viral test. Typhoid viral test. Typhoid. Viral test typhoid, viral test typhoid. Hemozoin, plasmodium, hemozoin, plasmodium. Hemozoin, plasmodium, plasmodium. Common cold, rhinovirus, common cold, rhinovirus, common cold, rhinovirus. Rhinovirus, common cold. Rhinovirus, common cold. Rhinovirus, common cold, rhinovirus. Allergy, dust and mites. Allergy. Physical barrier, mucus skin on our body is the main barrier. On our body is the main barrier which prevent entry of microorganisms. Gastrointestinal and urinary physical barrier. Epithelial lining respiratory. Gastrointestinal, urogenital tract helps in trapping microbes entering our body. Physiological barrier, acid in stomach, saliva in mouth, tears from eyes, microbial growth. Physiological barrier, acid in stomach, saliva in mouth, tears from eyes. Cellular barriers, certain types of leukocytes, polymorphonuclear leukocytes, Amino neutrophils, and monocyte, and the natural killers, types of lymphocyte in blood, as well as macrophages and tissues can phagocyte and destroy microbes. Non-mediated IUD. Non-mediated, okay, that is on lipase loop. Non-mediated, MEDIATED, non-mediated lipase loop, LIPPS lipase loop, and copper releasing IUD. Copper T, multi-load, 375, okay, 378, I forgot, 375, okay. Copper T, multi-load 375. Then hormone releasing IUD. In plants. Progestogen, okay. And then the 20. Barrier, diaphragms, walls, diaphragms, and cervical caps. Intrauterine devices. Non-mediated lipase loop. non-mediated है ना, non-medicated है, so non-medicated lipase loop. Okay, copper releasing IUDs, copper T, copper 7, multi-load 375, copper T, copper 7, and multi-load 375, multi-load 375, lipase loop, copper releasing IUDs, that's 100. These intrauterine devices are presently available as the non-medicated IUDs, lipase loop, copper releasing, copper T, copper 7, copper T and copper 7, copper T, copper 7, and multi-load 375, hormone releasing IUD, LNG20, and progesterone. LNG, LNG20, progesterone and LNG20. IUD increases phagocytosis of sperm within the uterus. Within the uterus, okay. Copper ion releasing suppress the motility of sperm and make cervical hostile. ना यूएस को इनर सो भगवान, या अल्लाह मेरे को बचा लीजिए आप. Copper ion released suppresses sperm motility and fertilizing capacity of sperm. Reduces fertilizing capacity, suppress sperm motility. Okay, hormone releasing IUDs in addition make the uterus unsuitable for implantation and cervix hostile to sperm. Sperm motility and with fertilizing capacity of reduces fertilizing capacity of sperm. The hormone releasing IUDs in addition makes the uterus unsuitable for implantation and cervix hostile to the sperm. IUDs ideal contraceptive. IUDs ideal contraceptives. You don't want to take pregnancy or space children. It is one of the most widely accepted methods of contraception in India. Implants का क्या था, मैं पूरा भूल गई यार. Progestogens alone. Progestogen alone or combination with estrogen can also be used by female as injections or implants under the skin. The mode of action is similar to that of pills and their effective periods are much longer. Okay. Implants. Pills को जैसे काम गर्छ progesterone. Okay. Progestogen, estrogen implants, okay. Hormone releasing IUD. Progesterone. Non-mediated. Lipase loop. Non-mediated lipase loop. Copper releasing IUD. CU7 multi-load. Copper releasing multi-load 375, okay. Hormone releasing IUD, LNG20. Hormone releasing IUD20 or progesterone. Implant progestogen implants, okay. आज पढ़ो कल भूल जाओ, क्या फायदा इससे। Following are the stages of cell division. Choose the correct sequence of stages from the option given below. Okay. First gap 1, E, then synthesis EC. भाई साहब इतना गंदा किया था क्या मैंने। गैप 2 A, देन ये क्वेश्चन मैंने एग्जाम में भी गलत किया था। देखो देखो, इसको हम लोग ना ऐसे याद करते हैं। गीतू नाम की एक लड़की है, पहले भी मैंने बताया था। वो एस पोकेमॉन के एस से लेस पावरफुल है या फिर पीछे आती है क्योंकि उसके पास पोकेमॉन नहीं है। तो G1 S एंड देन G2 दैट इज G2। गैप 1, देन इजी तो है भाई साहब, देन C सिंथेसिस, देन गैप 2 ECA। ECA यहीं पर दे, ओ फिर भी मैंने सही किया था। ECA उसके बाद में गैप 1 सिंथेसिस, गैप 2, उसके बाद आता है माइटोसिस। उसके बाद इस सेल डिवीजन आएगा। पहले कार्डियोकाइनेसिस आएगा, उसके बाद साइटोकाइनेसिस आएगा ना। देखिए डी, उसके बाद बी, उसके बाद बी, CECA DD। हाँ ऑर्डर गैप 1 S G2 M phase and finally G2। okay, G1 S G2 and this is cytokinesis last one। भाई साहब ये मैं इसमें गलत करके आई। रोज पढ़ना लिपिस लूप 3 इको 3 एक 3 दिया तो है, फिर मैंने गलत किया है। प्रोजेस्टोजन एंड प्रोजेस्टोजन एस्ट्रोजन ये भी हो सकता है। PBU 3 to 2 PBU 3 PBU 322 PBU है ना, PBR और PBR 322 PBR 322 okay। The following diagram shows a restriction site in E. coli cloning vector PBR 322। Find the role of X and Y। The following diagram shows a restriction site in E. coli। X and Y chain, okay। BAMH1 SAL1 ampicillin resistant हो यो AMPR ampicillin resistant right यो साइड का ampicillin resistant है होइन tetramycin resistant tetramycin BAM SAL BAMH1 SAL1 कस्तो परवीन AMPR ampicillin resistant ये तो TET tetramycin resistant ampicillin मैं गुस्सा ampicillin R ampicillin resistant tetracycline or kanamycin resistant okay tetracycline BAMH1 SAL1 PST1 PVU1 eco R1 CLA1 HIND3 CLA1 eco R1 PVU1 PST1 ampicillin resistant tetramycin resistant kanamycin BAMH1 र SAL1 टेट्रा मैं बार भूल जाती हूँ एक ही चीज बुढ़वा कलर की tetramycin resistant ampicillin resistant 80 ऐसे 80 एट तो नहीं लिखोगे ना तुम ता हो गए तो एट लिखना है एट ampicillin resistant tetramycin resistant okay say PVU Y proteino XQ origin of replication. The gene X is responsible for controlling the copy number of the linked DNA and Y for the protein involved in the replication of plasmid. Okay, good. ROP PVU2 ORI origin of replication. The gene X is responsible for controlling the copy number of the DNA. ORI and ROP. Okay. फिगर ले आते हैं ampicillin resistant TET tetramycin resistant okay BAMH1 then SAL1 BAMH1 SAL1 okay PVU1 PST1 PVU1 and PVU1 Praveen Past ORI controlling the copy number ROP PVU2 ROP ROP okay ROP PVU2 okay यहाँनिर तीनटा कुरा छ एउटा त हो हिन्ड 3 देन अर्को हो हाम्रो इको R1 हिन्ड 3 इको R1 र अझै एउटा छ के हो हिन्ड 3 इको R1 एन्ड हिन्ड 3 इको R1 कैसे भूल जाती है यार तो अभी CLA1 CLA1 हिन्ड 3 इको R1 PUV1, PUVN1, PST1. Praveen first. PVU1, PST1, ampicillin resistant. Okay. ORI, origin of replication, ROP. PVU2, ROP, ROP, PBR 322. Okay. Oh, PBR 322. PBR 322. E. coli cloning vector के को E. coli को के E. coli cloning vector PBR 322. Cloning vector PBR 322. E. coli को cloning vector E. coli Salvan. BMH1 Salvan tetramycin. Protein inversion replication of the plasmid. Why? For protein involved in the replication of plasmid. Exactly same चीज लिख के दिया ROP. Group codes. ROP codes for protein. Codes for protein involved in the replication of the plasmid. Okay. In the replication of plasmid. This is for antibiotic resistance. मिसोजोई केरा वाला नहीं पता, मैंने नहीं किया है तो मैं क्यों करूँ? Choose the correct statement given below regarding juxtamedullary nephron. Loop of Henle of juxtamedullary nephron runs deeply into medulla. Juxtamedullary nephron outnumbers. Mitochondria and chloroplast are both double membraned bound. Inner membrane of mitochondria is relatively less permeable as compared to chloroplast. Substrate enzyme complex for free enzyme ready to bind with another substrate. Bone marrow is the main lymphoid organ where all blood cells, including lymphocytes, are produced, correct? Both bone marrow and thymus provide microenvironment for development and maturation of T lymphocyte. खुद का क्या करूं? The human immune system consists of lymphoid organ, tissues, cell and soluble molecules like antibodies. As you have read, immune system is the unique in the sense that it recognizes foreign antigens, responds to this and remembers them. The immune system also plays an important role in allergic reaction, autoimmune disease and organ transplantation. Lymphoid organ. These are the organ where origin and or maturation and proliferation of lymphocyte occurs. The primary lymphoid organ where immature lymphocyte differentiate. So microenvironment for development and maturation of T lymphocyte. यार Bone marrow is the main lymphoid organ. Bone marrow is the main lymphoid organ where all lymphoid organ where all the blood cells, including produced. Bone marrow is the main lymphoid organ where all the blood cells, including lymphocytes, are produced. Bone marrow and thymus provide microenvironment for development and maturation of T lymphocyte. The thymus is a lobed organ located near the heart and beneath the breastbone. शिट, तो जोर से भी नहीं खींचना था मुझे, सारा गुस्सा मैंने रबर बैंड पर ही निकाल दिया. ₹20 पानी में गई, छपाक हो गया मेरा. Bone marrow is the main lymphoid organ where all the cells, including lymphocytes, are produced. Correct? Okay. Both bone marrow and thymus. ऐसा तो कोई statement है और यहाँ पर है तो both bone marrow and thymus provide microenvironment for the development and maturation of T lymphocyte. Both bone marrow and thymus provide microenvironment for the development and maturation of T lymphocyte. Statement 1 and statement 2 is both statement 1 and statement 2 are correct. एकदम यहीं से आ गया था, फिर भी मैंने नहीं लिखा. Both bone marrow and thymus provide microenvironment for development and maturation of T lymphocyte. Thymus provide microenvironment for development and maturation of lymphocyte. Statement based. Okay. हाँ हो गया पागल. Unicellular salivary gland, compound epithelium नहीं वो वहाँ से. The cerebral hemisphere connected by nerve tract known as corpus callosum, ये भी वहीं का है. RNA polymerase 3. और termination of transcription हो गया ये है ना, सही है. Splicing of exon, snRNA. टाटा बॉक्स प्रमोटर के साथ था आई थिंक. प्रमोटर डिको टू. Okay. Termination of cofactor. Termination of transcription BCO 3. RNA 3 snRNA tRNA. RNA polymerase 2 hnRNA heterogeneous. भाई साहब पागल हो जाऊंगी मैं. जैसे दिया भी नहीं होगा उसमें. Gauss competitive exclusion. Gauss competitive exclusion. two closely related species competing for different resources cannot exist. Different? Gauss' competitive exclusion principle states that two closely related species competing for different resources cannot exist indefinitely. Gauss' competitive exclusion principle, इसने क्या बोला था देखते हैं. States that, okay, Gauss' competitive exclusion principle states two closely related species competing for same resources cannot coexist indefinitely. And true or false? According to Gauss' principle, during competition, the inferior will be eliminated. This may be true if resources are limiting. This statement is true. कैसे तो ऐसा कर सकते हैं? Competitive. During competition. कहाँ गया during competition वाला? During competition, the inferior will be eliminated. Inferior one will be eliminated eventually. This may be true if resources are limiting, but not otherwise. Okay. 2024. According to Gauss' principle, during competition, the inferior will be eliminated. Inferior one will be eliminated. This may be true if resources are limiting. During competition, the inferior one will be eliminated if the resource. During competition, the inferior will be eliminated. True if resources are limiting. Statement 1 is false, but statement 2 is true. ये वाला होगा. Gauss' competitive exclusion principle states that two closely related species competing for same resources cannot exist indefinitely. Two related species competing coexist indefinitely. Same resource. Same था यहाँ पर differently दिया ऐसे. गुस्सा तो आता है लेकिन क्या करूँ? कंट्रोल बहन कंट्रोल. एक लो में खा ले. The structure used for storing food particle. Gizzard helps in grinding the food particles. Gizzard, the structure used for grinding the food. Okay, gizzard कहाँ गया? Gizzard helps in grinding the food particles. Gizzard helps in grinding the food particles. Gizzard, the structure used for grinding food. Gizzard helps in grinding the food particle. This is PYQ. D को 1 हे तीनों में था. Crop. Crop. This in turn opens, esophagus, narrow tubular passage called esophagus. This in turn opens into a sac-like structure called crop, used for sac-like structure used for storing food. Crop को A को 4. Crop. Sac-like structure. It is a sac-like structure. बोरा जस्तो हुँदो रहेछ के crop. Sac-like structure called crop used for storing food. PYQ. Use for storing food. PYQ use for storing food. Crop. Anal circuit. क्यों anal circuit के not anal this time. Anal circuit. Joint filamentous structure. 10th segment. Ring of 6 to 8 tubules at junction of foregut and midgut. Ring of 100 to 150. Yellow colored thin filaments at the junction of midgut and hindgut. Gastric secant malpighian tubule. At the junction. कल लेके आऊंगी ना एक. At the junction, the midgut and hindgut. At the junction of midgut and hindgut. At the junction of midgut and hindgut is present another ring of 150 to 100 to 150. Ring of 100 to 150. At the junction of midgut and hindgut. At the junction of midgut and hindgut is present another ring at the junction of midgut and hindgut. Another ring of 100 to 150 yellow colored thin filaments. This malpighian tubules. At the junction of the midgut and hindgut. ये वाला तुम्हारा gut रहेगा पूरा. Foregut, okay. midgut, hindgut. At their junction, hindgut is present another ring. एक और रिंग रहेगा. Ring, another ring. 100 to 150 yellow colored thin filament. Midgut, hindgut, junction, ring. 100 to 150 tube, 100 to 150 yellow colored thin filamentous Malpighian tubules. Of 150 100 to 100, 100 to 150 yellow colored thin filamentous at the junction of midgut and hindgut Malpighian tubule. Seco 3, Deco 2, gastric CK. Ring of 6 to 8 blind tubules. 6 to 8. Ring of 6 to 8 blind tubules called hepatic or gastric CK. Cica. Function of foregut and midgut. Gastric cica. Okay, hepatic or gastric cica. Hepatic or gastric cica. ओके, तो यहाँ पर भी होता है 1 2 3 4 5 6 1 2 3 4 5 6. Okay. At the junction of midgut and hindgut is present another ring of 100 to 150 yellow colored thin filament Malpighian tubules. 6 to 150. A ring of 6 to 8 blind tubules called hepatic or gastric CK. Gastric, GASTRIC, gastric CEA. Not CAE. CAE. Hepatic or gastric. Storing food. Crop. A ring of 6 to 8 blind tubules at junction of foregut and midgut. Gastric. Cica. Gizzard. Structure used for grinding food. Structure used for storing food. Crop. Foregut or midgut को बीच मान सके. Okay. Heart muscles are electrically silent. Depolarization of ventricle. Depolarization of atria, repolarization of ventricle, T wave. Depolarization of atria, P, ventricle, Q, R, S. Q, R, S. TP gap. TP gap, T और P के बीच के gap. P wave represents depolarization. P wave represents depolarization of the atria. PYQ. QRX complex, depolarization of the ventricles. QRS, depolarization of the ventricles. T wave repolarization, कहाँ गया T wave? यहाँ पर है. T wave represents repolarization of ventricle. Repolarization. T wave represents ventricle repolarization, because depolarization of the ventricle represents presence of atria. then उसके बाद हम लोगों को दिया है TP gap. TP gap, ये TP gap का तो नहीं दिया है ना. Depolarization of ventricle, COO4, ACO3, ACO2, DCO1. Heart muscles are electrically silent. ऐसा कुछ नहीं दिया है. TP gap. TP gap. Heart muscles are electrically silent. As per ABO blood grouping system, the blood group of father is B positive and mother is E positive. As child is O positive, the respective genotype can be. ये तो इजी है. तो फादर का आई, फादर क्या है? बी पॉजिटिव ना, अम्मा है ई पॉजिटिव और बच्चा है ओ पॉजिटिव. आई आई है, आई ए आई है, आई बी आई है, आई बी आई, आई ए आई, यही तो है, ए करेक्ट है, नहीं आएगा। Identify the correct action A, B, C, D with respect to spermatogenesis. Interstitial cell, B, interstitial cell. भाई साहब, अस्तगफिरुल्लाह, ladies cell interstitial, luteinizing hormone, androgens, formation of spermatids. Okay. should be का ladies cell, ladies cell. इस तो मैंने फर्स्ट में ही एलिमिनेट कर दिया। ये एलिमिनेट नहीं किया है अभी। ये वाला ये तो भगा दिया ना, FSH। अच्छा 3 वाला सही है, क्योंकि फॉलिकुलर गोनाडोट्रोपिन रिलीजिंग हार्मोन FSH को ही बोलते हैं यार। सोटोली सेल, सोटोली सेल को नहीं, लेडिक सेल को ही जाएगा ये। फिर भी ये वाईफाई जाता रहता है, मैं कुछ नहीं कर सकती। हाइलाइटर लग गया है। Excess secretion of cortisol, moon phase and hyperglycemia. Okay, moon phase and hyperglycemia ये तो नहीं पता, बट यहाँ से अपना इसका है, इसको बोलते हैं एक्रोमेगाली। एक्रोमेगाली हाँ दिया है तो, हाँ एक्रोमेगाली यही है। ओके, डिस्टॉर्शन ऑफ फेस बोला है ना। हाइपर सिक्रेशन ऑफ थायराइड हार्मोन एंड स्टंटेड ग्रोथ, हाइपोकम्पी। ओके, स्टंटेड ग्रोथ, दैट इज thyroid hormone, stunted growth, thyroid hormone, क्रिटिनिज्म भी बोल सकते हैं ना, थायराइड को ही क्रिटिनिज्म बोलते हैं। सीटी क्रिटिनिज्म है तो क्रिटिनिज्म। हाइपर सिक्रेशन ऑफ थायराइड हार्मोन एंड प्रोट्रूडिंग eyeball, ग्रेव्स डिजीज, एक्सोथर्मिक क्वेटर, सही तो लगाया है। एक्सेसिव सिक्रेशन ऑफ ग्रोथ हार्मोन, जो बचा है उसने लगा देना। देखो, एक्सोथर्मिक क्वेटर, हाइपर सिक्रेशन ऑफ थायराइड हार्मोन, 3 इको 3, तो ये भी गलत है, ये भी गलत है, ये भी गलत है। हाइपोथैलेमिक क्वेटर, एक्सोथर्मिक क्वेटर, हाइपर सिक्रेशन ऑफ थायराइड हार्मोन एंड प्रोट्रूडिंग आईबॉल, सही तो है। बी को 4, एक्रोमेगाली। देखो तो सही है। देखो 4 अच्छा कॉर्टिसोल, एक्सेसिव सिक्रेशन ऑफ कॉर्टिसोल बोला है। Excessive secretion of cortisol, moonfish and hyperglycemia.
Excessive cortisol secretion or hypercortisolism can lead to hyperglycemia, high blood sugar, because cortisol interferes with how insulin works, leading to impaired glucose metabolism and elevated blood sugar levels. Here's a more detailed explanation. 1. Cushing syndrome and hypercortisolism. Cushing syndrome is a condition caused by prolonged exposure to high levels of cortisol, which can be due to the body producing too much cortisol or from taking certain medications. 2. Cortisol's role. Cortisol, a stress hormone, plays a role in regulating blood glucose levels, but when present in excess, it can disrupt this regulation. 3. Mechanism of hyperglycemia. Excess cortisol impairs insulin's ability to lower blood sugar, leading to hyperglycemia. 4. Consequences of hyperglycemia. Hyperglycemia can increase the risk of developing diabetes and other complications. 5. Treatment. Managing hyperglycemia and Cushing syndrome often involves treating the underlying cause of hypercortisolism, which can help lower cortisol levels and improve blood sugar control. 6. Other factors. Other hormones like glucagon and catecholamines can also contribute to hyperglycemia. 7. Research. Studies have shown a correlation between cortisol levels and hyperglycemia in various conditions, including type 2 diabetes and Cushing syndrome. And 8. Moonfish. The term moonfish is not relevant to the medical context of excessive cortisol secretion and hyperglycemia. This is for informational purposes only. For medical advice or diagnosis, consult a professional. Generative AI is experimental.
Hypersecretion. Okay. Hyperthyroidism, hypersecretion of thyroid hormone, protruding eyeball, cretinism, hyposecretion of thyroid hormone, cretinism, hyposecretion. Okay. Hypothyroidism, stunted growth, cretinism. Stunted growth, cretinism, PYQ25. Hypothyroidism during pregnancy, stunted growth, cretinism, stunted growth, cretinism, stunted growth, cretinism. इतने जोर से मारा फिर भी नहीं मरा. Hypothyroidism during pregnancy, stunted growth, cretinism. hypothyroidism, exophthalmic hyperthyroidism of eyeball. Cretinism, Cushing syndrome, excess secretion of cortisol. cortisol कहाँ गया? यही तो होना चाहिए ना उसको. cortisol आउटर एनसीईआरटी था. वाह. cortisol. Secretion. Moon phase. Hyperglycemia. Cushing syndrome. Excess secretion of cortisol. Hyperglycemia. छोड़ो, लिख दिया है ना. स्मॉल ICSH क्या होता है? ICSH सुबह हो गया क्या? बर्ड्स तो चीयर करने लगे. ICSH ICSH हार्मोन, ल्यूटेनाइजिंग हार्मोन LH. अच्छा, उसको ICSH भी बोलते हैं. इंटरस्टिशियल सेल स्टिमुलेटिंग हार्मोन. थ्री हो गया है. मेरा तो कंप्लीट ही नहीं हुआ है. सोए कि थोड़ा पढ़े. Gonadotropin releasing hormone GnRH stimulate the pituitary synthesis and releases gonadotropins. On the other hand, GnRH बोला है ना, gonadotropin releasing hormone. देखो LH and FSH stimulate gonadotropins, stimulate gonadal activity, and hence are called gonadotropins. GnRs, LH and FSH, LH and FSH. फॉलिकल स्टिमुलेटिंग हार्मोन, इसको क्या बोला था? LH को ल्यूटिनाइजिंग हार्मोन को ही तो बोला था. फॉलिकल स्टिमुलेटिंग हार्मोन लेडिक सेल कर दिया मैंने. क्यों इतना ज़्यादा कंफ्यूज होती हूँ? ल्यूटिनाइजिंग हार्मोन LH, इसको क्या बोलते हैं? ICASH. इंटरस्टिशियल सेल स्टिमुलेटिंग हार्मोन, ल्यूटिनाइजिंग हार्मोन. LH and FSH stimulates gonadal activity and hence are called gonadotrophin. In males, LH stimulate and synthesis and secretion. Stimulates synthesis and secretion of hormone androgen. LH. Stimulate secretion and synthesis of hormone called androgens from testes. टेस्टिस दिया है बी में. सोटोली सेल, लेडिक सेल, इंटरस्टिशियल सेल दिया है. In female, luteinizing hormone induces ovulation in fully mature spermiogenesis. Okay. In male, FSH and androgen regulate spermatogenesis. अच्छा भाई, तू भी गलत है तो फिर. Spermatogenesis. Necessary. FSS along with C. Androgen दिया है. लेडिक सेल, सोटोली सेल, लेडिक सेल. भाई साहब ये क्या दिया है? मेरे को कंफ्यूज ही कर दे रही है. तो सेरेब्रल हेमिस्फीयर आर कनेक्टेड बाय नो. ये वाला करना है ना, ये भी करना है. स्पेशली ये तो करना ही होगा. बोन मैरो वाला कर लिया है, ये भी कर लिया है. सबस्ट्रेट एंजाइम कर लेंगे. ये भी करना है, ये भी करना है, ये भी करना है. The cerebral hemisphere are connected by nerve tract known as corpus callosum. The brain stem consists of the medulla oblongata, pons and cerebellum. cerebrum तो नहीं होगा, ये तो पक्का फॉल्स ही होगा. ब्रेन स्टेम यहाँ पर देना चाहिए. हाँ, ब्रेन स्टेम दिया है. The major reason make up the brain stem. 3 major reasons make up the brain stem. Midbrain, pons and medulla oblongata. इसमें क्या दिया है? मिडब्रेन की जगह pons and medulla oblongata. करेक्ट. मिडब्रेन की जगह में इन लोगों ने दिया है सेरिब्रम, सेरिब्रम फोर ब्रेन का पार्ट है ना, सेरिब्रम, हाँ गलत गलत। Exact, I have statement. The three major reasons make up the brain stem consists of midbrain, pons, and medulla oblongata. MPM. Midbrain, medulla oblongata, and pons. Okay. Statement based आया था ये. Three major brains can make up brain stem. Midbrain, pons, and medulla oblongata. Brain stem forms the connection between the brain stem forms the connection between brain and spinal cord. Brain and spinal cord. Brain stem forms the connection between brain and spinal cord. Brain and spinal cord. Brain stem. Medulla oblongata, pons, and midbrain. Brain stem. Midbrain, pons, and medulla oblongata. Pons and medulla oblongata. Statement 2 are incorrect. Okay. Statement 2 are correct. और correct लिख दिया भाई साहब. Cerebral hemisphere are connected by nerve tract. Connected by tract of nerve called corpus callosum. इन्होंने corpus callosum ही बोला है. देखो कहाँ गया. हाँ यहाँ पर. The cerebral hemisphere. ये मच्छर को ना मैं A deep cleft divides the cerebrum. Forebrain consists of cerebrum, thalamus, and hypothalamus. Thalamus, hypothalamus. Cerebrum consists of thalamus, hypothalamus. Cerebrum, thalamus, and hypothalamus. Cerebrum forms the major part of the human brain. Major part, cerebrum. A deep cleft divides the cerebrum longitudinally into two halves, which are termed as the right and left cerebral hemisphere. Cerebral hemisphere are connected. Cerebral hemisphere are connected by tract of nerve fiber. Connected by nerve tract known as corpus callosum. Corpus callosum. Okay, तो ये सही वाला था. Statement based. Cerebral hemisphere are connected by tract of nerve fiber called corpus callosum. Corpus callosum कनेक्ट करता था. क्या कनेक्ट करता था? corpus callosum. मुझे बहुत ज़्यादा गुस्सा आ रहा है, लेकिन मैं खैर कुछ कर भी नहीं सकती गुस्सा करने के अलावा ही. Statement 1 is correct, but statement 2 is incorrect होना चाहिए था. corpus callosum. इसे कैसे याद करी? अब इतना ज़्यादा टर्म है कि इसमें भूल ही जाती हूँ, कल ही भूल जाऊंगी ये. Cerebral hemisphere. Cerebral hemisphere are connected by nerve tract known as मच्छर के बच्चे. मरता ही नहीं है. Cerebral hemisphere are connected by nerve tract known as corpus callosum. True. Cerebral hemisphere are connected by nerve tract known as corpus callosum. The brain stem consists of the medulla oblongata, pons, and cerebrum. during comparison. Statement 1 and 2 are correct. Bone marrow is main lymphoid organ where all blood cells, including lymphocytes, are produced. Both bone marrow and thymus provide microenvironment for the development and maturation of T lymphocyte. Regarding catalytic cycle of an enzyme action, select the correct sequence. Okay, ये बायोमोलिक्यूल से कर लेना, ये भी कर लेना, ये भी कर लेना, ये भी कर लेना, इससे पहले ये कितनी वाला मुझे बिल्कुल पसंद नहीं है. Choose the correct statement given below regarding juxtamedullary nephron. अरे ये तो यहीं से देखो ना प्लीज. और है क्या juxtamedullary nephron? Juxtamedullary nephron 15% large. They are large. 15% large. Maximum part present in medulla. Maximum part present in medulla. Vasa recta present. Loop of Henle long. HL क्यों HL? अच्छा loop of Henle is loop long होगा. Walk during emergency. Loop of Henle of juxtamedullary nephron runs deep into medulla. They are present in medulla. सब में कहीं तो लिखा है यहाँ पर. Maximum part present in medulla. Loop of Henle of juxtamedullary nephron runs deep into medulla. Juxtamedullary nephron outnumber the cortical nephron. ना वो 85% होता है. Cortical nephron 85%. Juxtamedullary nephron only 15%. Only 15%. मच्छर कहाँ से आ जाती है? Juxtamedullary nephrons are located in the column of Botany. Column of Botany? मेडुला में होते हैं. मैक्सिमम पार्ट प्रेजेंट इन मेडुला के कॉर्टेक्स में आंसर. कॉलम ऑफ बॉटनी. हाँ कॉलम ऑफ बॉटनी वो वाला बीच का. मेडुला में होता है मेडुला. रीनल कॉर्पस्कल ऑफ juxtamedullary nephron. Renal corpuscle lies in the outer portion of the renal medulla. क्या क्या होगा है इन लोगों में. In some of the nephron, the loop of Henle is very long, runs deep into the medulla. This nephron are called juxtamedullary nephron. यहाँ पर ये है तो देखो. The loop of Henle is very long, runs deep into the medulla. They runs deep into the medulla. Medullary nephron. These nephrons are called loop of Henle of juxtamedullary nephron runs deep into the medulla. Loop of Henle is very long and runs deep into the medulla. Juxtamedullary nephron. following are the statement about non-chordate. Okay, non-chordate. Pharynx is perforated by gill slit. Notochord is absent. So you. Central nervous system is dorsal. Ventral. Ventral. Heart is dorsal if present. Postnasal tail is absent. मुझे तो पता भी नहीं, मैं पूरा भूल गई हूँ, फिर से देखना पड़ेगा. Chordate, notochord present. Okay, chordate, notochord present. Central nervous system is dorsal, hollow, and single. Non-chordate, notochord absent. Okay. Central nervous system in non-chordate is ventral, solid, and double. Ventral, solid, and double. Non-chordate. Pharynx perforated by gill slit. Chordate. Gill slits are absent in non-chordate. Heart is dorsal, पिछाड़ी पट्टी हुन्छ if present. हाम्रोमा के हुन्छ heart is ventral. Post anal tail is present. हाम्रोमा छ, उनीहरुमा के हुन्छ absent हुन्छ non-chordate मा. Okay, thank you. Notochord present. हाम्रोमा के हुन्छ chordate मा notochord present हुन्छ. Dorsal post anal tail is absent. Gill slits are absent. Central nervous system double. Notochord present. Okay. Following are the statement based. Chordate, notochord present. Chordate, notochord present. Heart ventral huncha. Central nervous system dorsal, hollow, and single. Dorsal, hollow, and single huncha. Notochord present huncha. Chordate ma notochord present. Central nervous system dorsal, hollow, and single. Pharynx perforated by gill slit. Pharynx ke huncha perforated by gill slit. Heart is ventral. Post anal tail is present. Heart is ventral. ये याद हो जाएगा ना. Post anal tail is present. Pharynx perforated by gill slit. Notochord present. सब कुछ ही होता है हम लोगों के पास. Notochord भी होता है. Pharynx perforated by gill slits. Post anal tail is present. Heart is ventral. CNS is dorsal. hollow and single. Notochord present. Poststernal tail present. Central nervous system dorsal hollow and single. Then heart is ventral. Gill slits are perforated by pharynx perforated by gill slit. देखते हैं कैसा होगा. Pharynx perforated by gill slit. Pharynx perforated by gill slit images. Mammals pharynx. In mammals, the pharynx or throat, a muscular tube connecting the nasal and the oral cavity, larynx and esophagus. Okay. Do humans have perforated pharynx? Well, pharyngeal perforation, a hole or tear in pharynx, is rare. It can occur in human, often due to atrophic cause like instrumentation or blunt trauma. Pharynx perforation is a tear or hole in the wall of the pharynx, as known as the throat. Pharynx images. Pharynx. Oropharynx. Okay, pharynx. Pictures of perforated pharynx. Pictures of perforated pharynx. Fringes named pharynx, bro. Pharynx. PHARYNGX. Perforated pharynx. ये हम लोगों के इंटरनल बॉडी इतना डिस्गस्टिंग क्यों दिखता है दिखने में. Anyways, CNS, central nervous system, is dorsal hollow and single. Pharynx perforated gill slit. A notochord is present. Oh, show me the picture of notochord. Notochord is present. Pharynx perforated gill slit. A notochord is present. CNS, central hollow and single. Poststernal tail is present. One more is there. Heart is ventral. This is notochord. Poststernal tail. Poststernal tail in chordates. छोड़ो इसको मत देखो. Non-chordates. Postantral tail is absent. Notochord is absent. CNS is ventral, solid and double. Absent other than this. Heart is dorsal if present. Gill slits are absent. Okay, which of the following statement about non-chordates? The following are the statement about non-chordates. Choose the most appropriate answer for the option given below. Notochord is absent, correct. Pharynx is perforated by gill slit. Nah, false. Non-chordates and the non-chordates now. False. Pharynx is not perforated by gill slit. Gill slit it's absent होता है. Notochord is absent, correct. True. इन लोग सच में absent बन सके. हम लोग present है notochord. Central nervous system is dorsal. CNS is dorsal. True. Heart is dorsal if present. True. Postantral tail is absent. True. B, D, E. B, C, D, E. भाई मैंने कैसे डॉर्सल वेंट्रल होता है ना इन लोगों का अभी तो पढ़ा वेंट्रल हॉलो बी डी ई करेक्ट ये भी हो गया नरो वाला हो गया ये वाला हो गया अभी तो क्या है माइटोकॉन्ड्रिया एंड क्लोरोप्लास्ट वाला Mitochondria and chloroplast. Mitochondria and chloroplast both are double membrane bound organelles. Double membrane bound ही तो होता है हाँ सही है Mitochondria is a double membrane bound structure. PYQ 2024. Is it statement based? Yes, it is. Structure. Inner membrane of mitochondria. Okay, with the two outer membrane and inner membrane dividing its lumen distinctly into two aqueous compartment, the outer membrane and inner membrane compartment. The inner compartment is filled with a dense homogeneous substance called the matrix. The inner compartment is filled with a dense homogeneous substance called the matrix. The outer membrane forms the continuous limiting boundary of the organelles. The inner membrane forms a number of enfoldings called मुझे ऐसा क्यों लग रहा है कि कोई भूत रूम में घुस गया है मेरे अलावा यहाँ पर कोई नहीं है तो मैं शायद खुद को ही भूत बोल रही हूँ The inner compartment is filled with a dense homogeneous. अगर मैं ना लाइव आके नहीं पढ़ रही होती अकेले पढ़ रही होती ना तो मैं बहुत डर जाती अभी लेकिन अभी मैं नहीं डर रही क्योंकि मेरे साथ लोग हैं ना The inner compartment is filled with a dense homogeneous substances called the matrix. The outer matrix forms the continuous limiting boundary of the organelles. The inner membrane forms a number of enfoldings called the cristae towards the matrix. The cristae increase the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Chloroplast contain chlorophyll and carotenoid pigment which are responsible for trapping light energy essential for photosynthesis. In chromoplast, fat-soluble carotenoid pigments are like carotene, xanthophyll and other are present. This gives the part of the plant a yellow, orange or red color. The leucoplasts are the colorless plastids of varied shape and size with the stored nutrients. Leucoplast stored nutrition. Chloroplast chlorophyll containing pigments. Chlorophyll and carotenoid pigment of chloroplast pigments. Chromoplast fat soluble carotenoid. A myeloplast stores carbohydrate, example potato. Elaioplast stores oil and fats, whereas a leuroplast stores protein. Majority of chloroplast of the green plants are found in the mesophyll cell of the leaves. (20 to 40% in mesophyll. Double membrane like mitochondria. Exactly यही था ना, mitochondria and chloroplast both are double membrane bound organelle. Mitochondria and chloroplast are double membrane bound. Okay, PYQ. Statement based वाला था. और दूसरा वाला क्या था? Inner membrane of mitochondria वाला था ना. Of the two, the inner chloroplast membrane is relatively less permeable. Inner membrane of mitochondria is relatively less permeable as compared to chloroplast. Now, of the two, mitochondria and chloroplast, inner chloroplast membrane is relatively less permeable. This is false. Oh, good job. तुमने कितना अच्छे से किया है. I'm so proud of you. Mitochondrial double membrane. Okay. Of the two, inner chloroplast membrane is relatively less permeable. Inner chloroplast membrane is relatively less permeable in comparison to mitochondria. Chloroplast. CHLOROPLAST. Chloroplast is relatively less permeable as compared to mitochondria. यहाँ पर होता है. अल्हम्दुलिल्लाह ये भी तो करना है. इसमें नहीं दिया है. आई डोंट थिंक इसमें दिया है. मॉर्फोलॉजी ऑफ फ्लावरिंग प्लांट नहीं, थर्ड इससे पहले तो होना चाहिए ना. एनिमल किंगडम. एनाटॉमी ऑफ फ्लावरिंग प्लांट की भी बाद में है. ओके, अच्छा ये पहले कर लेते हैं ना. Regarding catalytic cycle of an enzyme action, select the correct sequential step. Substrate enzyme complex. ये रहा तुम्हारा substrate. Enzyme substrate enzyme complex is for substrate enzyme complex formation. Okay, free enzyme ready to bind with. इसके बाद होगा एंजाइम. आगे फ्री, यहाँ पर होगा प्रोडक्ट. ये रहा तुम्हारा एंजाइम. फ्री एंजाइम इसके बाद ही आएगा. फ्री एंजाइम रेडी टू बाइंड विद अनदर सबस्ट्रेट. ये लास्ट वाला है, मतलब कि ABCD E. ये वाला E है तुम्हारा और ये वाला तुम्हारा D. ओके ना, ये वाला C है. ये वाला रिलीज ऑफ प्रोडक्ट D प्लस C है. Chemical bonds of the substrate broken. Chemical bond of the substrate broken. हाँ ये B है. Substrate binding to active site ये A है. ओके, मतलब कि सबसे पहले आएगा तुम्हारा E उसके बाद आएगा तुम्हारा डी, उसके बाद आएगा तुम्हारा ए, सी, देन फाइनली बी आएगा। ओके, सबस्ट्रेट बाइंडिंग टू एन एक्टिव साइट, केमिकल बॉन्ड्स ऑफ द सबस्ट्रेट ब्रोकन डाउन। मैंने ना ये पिछले वाले एग्जाम में मैंने ये इजी वाला क्वेश्चन छोड़ के आई थी, अभी मैंने देखा ये बिना पढ़े भी कोई भी कर सकता है, मैं छोड़ के आई थी तब, क्योंकि 15 चूज करना था ना। सबस्ट्रेट बाइंडिंग टू एक्टिव साइट, chemical bond of the substrate broken, chemical bond of the substrate broken, substrate enzyme complex formation, free enzyme ready to bind with another substrate, release of product, free enzyme ready to bind with another substrate. Okay, EDA. First, the substrate, okay, the catalytic cycle of an enzyme action, following steps. Okay, first, the substrate bind with the active substrate binding to active site, substrate binding to active site, 1 A. The binding of substrate induce the enzyme to alter its shape, fitting more tightly around the substrate. The active site of the enzyme, now in close proximity of the substrate, breaks the chemical bond of the substrate and chemical bond of substrate broken. Chemical bond substrate. The active site of the enzyme now is in close proximity of substrate. Active site of enzyme now is in close proximity of the substrate, breaks the chemical bond of the substrate. Okay. Chemical bond. Okay. D. ये क्या है? Substrate enzyme complex formation. Binding of substrate induces the enzyme to alter itself, fitting more tightly around the substrate. 3. उसके बाद 4. यहाँ तो कितना ज़्यादा दिया है? The enzyme releases the product. Enzyme releases the product. Of the reaction, and the free enzyme is ready to bind with another molecules of the substrate and runs to the catalytic cycle. A free enzyme is ready. Free enzyme is ready to bind. With another substrate. Last time, okay. 3. Here I have 4. DCB. DCD यही तो रहा है। ई के बाद ए है। ओके, Binding of substrate. Substrate binds to active site. Substrate binds to active site. Substrate enzyme complex formation. Substrate enzyme complex formation. Substrate enzyme. Substrate enzyme complex formation. Substrate enzyme complex formation. 1, 2, 3, 4, 5. Okay. Substrate enzyme complex formation. Substrate enzyme complex formation then breaks the chemical bond of the substrate. Breaks the chemical bond of the substrate broken. Chemical bond of the substrate broken. Substrate. Okay. Substrate bind to an active site. Substrate enzyme complex formation. Substrate enzyme complex formation breaks the bond of the substrate. Chemical bond of the substrate broken. Release of the product. Free the enzyme ready to bind with another substrate. Enzyme release. ये PYQ था सब कुछ. PYQ. Enzyme release from the product and ready to bind with the substrate. Excuse me. Buccal cavity, salivary gland. Okay. Unicellular glandular epithelium, compound epithelium, multicellular glandular, endocrine glandular epithelium, multicellular glandular epithelium. मुझे नहीं पता. मेरा लास्ट एक क्वेश्चन बचा है. मैं ये करके ना अच्छे से सोऊंगी. फुस करके सोऊंगी. पोर्शन हटा दिया है ये एनसीईआरटी वाले ने, और यहाँ से क्वेश्चन आया भी था। ये क्या बात हुई, यहाँ से क्वेश्चन आया था यार। यूनिसेलुलर ग्लैंडुलर एपिथेलियम, आई थिंक गॉबलेट सेल ऑफ एलिमेंट्री कैनाल, एको 3। हाँ तो ये तो ऐसे ही आंसर आ गया था। कंपाउंड एपिथेलियम मल्टीसेलुलर, कंपाउंड एपिथेलियम मल्टीसेलुलर ग्लैंडुलर एपिथेलियम, सलाइवरी ग्लैंड मल्टीसेलुलर था। हाँ okay, some of the columnar and cuboidal cells get specialized for secretion and are called glandular epithelium. Okay, they are mainly of two types: unicellular consisting of isolated glandular cell, goblet cell of alimentary canal, isolated, okay, and multicellular consisting of cluster of cell, multicellular glandular epithelium, salivary gland. Multicellular glandular epithelium, glandular epithelium. Oh, some of the columnar or cuboidal cells get specialized for secretion and are called glandular epithelium. They are mainly of two types: unicellular consisting of isolated glandular cell, goblet cell of the alimentary canal. Glandular cell ही तो दिखाया है यहाँ पर, तो glandular ही दिया है. Endocrine glandular epithelium, pancreas. Compound epithelium, moist surface of buccal cavity. Compound epithelium, compound epithelium, moist surface of buccal cavity. अच्छा, ये एक आउट ऑफ एनसीईआरटी हो गया क्या, नहीं पता मुझे. एंडोक्राइन ग्लैंड्स, एंडोक्राइन ग्लैंड्स कहाँ गया? Endocrine glandular epithelium, endocrine glandular epithelium, endocrine gland. Compound epithelium, compound epithelium, moist surface of buccal cavity. Moist surface of buccal cavity. I think वो वाला ईयर का तो सबसे गंदा था ना, PYQ. Compound epithelium, moist surface of buccal cavity. Compound epithelium, moist surface of buccal cavity. Pharynx, inner lining of duct of salivary gland, inner lining of ducts of salivary gland, and pancreatic duct. Pancreatic ducts. Okay. Compound epithelium is made up of more than one layer, multi-layered. Compound epithelium is made up of more than one layer, multi-layered, and thus has limited role in secretion and absorption. Multi-layered has limited role in secretion and absorption. Their main function is to provide protection. Main function to provide protection against chemicals and mechanical stress. Protection against chemical and mechanical stress. They cover dry surface of skin. Dry surface of skin. Dry surface of skin. Buccal cavity. Buccal cavity. Moist surface of buccal cavity. Dry surface of skin. Moist surface of buccal cavity. Dry surface of skin. Moist surface of buccal cavity, and then lining the ducts of salivary gland. Pharynx, even pharynx, buccal cavity, pharynx, both buccal cavity and pharynx. Inner lining of ducts of salivary gland. Inner lining of ducts of salivary gland. Salivary gland को inner lining में. Inner lining of ducts of salivary gland and pancreatic ducts Okay. Compound epithelium, moist surface of buccal cavity. जो मेरा chapters last year strong था वो इस बार नहीं है, अब तो मुझे पता चल गया. Compound epithelium, moist surface of buccal. लेकिन इस बार तो better करना था ना last year से. ये क्या हो रहा है मेरे साथ? Compound epithelium, moist surface of buccal cavity, moist surface of buccal cavity, moist surface. Okay, म भन्छु. Compound epithelium कहाँ पाउँछ भन्दाखेरि moist surface of buccal cavity, pharynx, inner lining of salivary duct, dry surface of skin, Ani, ducts of pancreas. They cover the dry surface of skin, moist surface of buccal cavity, pharynx, and inner lining of ducts of salivary gland and pancreatic ducts. Okay, हो गए 4, 1 2 3 right now. Dry surface 1, buccal cavity, pharynx से 3, inner lining of salivary gland 4, and finally pancreatic duct 5. Compound epithelium. Okay, compound epithelium, moist surface of buccal cavity. Then we have unicellular glandular epithelium. Unicellular glandular epithelium, goblet cell of alimentary canal. देखो तो यहाँ पर है, कहाँ गया? Unicellular, consisting of isolated glandular cell, goblet cell of alimentary canal. Unicellular, consisting of isolated glandular goblet cell of alimentary canal. Unicellular glandular epithelium. Compound epithelium भी हो गए. Multicellular glandular epithelium. Okay, some of the columnar givrils get specialized for secretion. Glandular epithelium. Multicellular, consisting of cluster of cells. Salivary gland. Okay, salivary gland. Multicellular. Multicellular glandular epithelium. Salivary gland. A को 3, B को 4. Salivary gland. Okay, multicellular and glandular epithelium ही था. And finally endocrine gland, glandular epithelium. On the basis of mode of pouring of the secretion, glands are divided into 2 categories: exocrine and endocrine glands. Exocrine glands secrete mucus, saliva. Exocrine gland secretes mucus, saliva, earwax, oil. Exocrine होता है ये सब. अच्छा. On the basis of mode of pouring of their secretion, glands are divided into 2 categories, namely exocrine and endocrine glands. Okay, exocrine glands. Exocrine glands secrete mucus, saliva, earwax, oil, milk, digestive enzymes, and other cells product. These are released through ducts or tubes. In contrast, endocrine gland do not have ducts. They do not have ducts. Their product called hormones are secreted directly into the bathing the gland. Okay. एंडोक्राइन ग्लैंडुलर एपिथेलियम. अब एक ही बचा है तो यही तो होगा ना, पैनक्रियास. एंडोक्राइन ग्लैंडुलर एपिथेलियम. एंडोक्राइन पैनक्रियास. हाँ, यहाँ पर भी तो दिया था ना इसका. यहीं पर है, देखो पैनक्रियास. Endocrine glandular epithelium. इसको ना पैनक्रियास देखो. Endocrine glandular epithelium. Endocrine glandular epithelium. सो जाती हूँ आप, है ना. वैसे लोग भी काफ़ी कम हो गए. ये बात हुई ना, अभी हम लोग चैट्स भी ऑन कर सकते हैं. पता नहीं कितने लोग भाग गए होंगे. मेरे सारे दोस्त लोग भाग गए क्या, लगता तो है. हाँ, मैं तेरा ही वेट कर रही थी शुभम, नॉइस लिखने के लिए. गए. देख लेंगे तो देखना है, देख लेंगे आगे का। डर के कुछ नहीं होगा, डर के आगे जीत है, और जीत के आगे मेडिकल कॉलेज। अकेले बोलने की आदत हो गई है मुझे शायद। हम लोग बचपन में ऐसा करते थे, किसी को पता है? कोई टीचर आए ना या ना आए, हम लोग जोर जोर से गुड मॉर्निंग टीचर, वो कोरस में होता था ना, तो गुड मॉर्निंग मूड गॉर्निंग एक ही सुनता था। मुझे ना ये पिलो कवर फेंक देना है, दूसरा वाला लगाना है, फेंक देना है, धोना है, कुछ भी मतलब कुछ भी बोलते हो। पिलो कवर चेंज करते ले रहना तुम लोग भी, ओके? अगर हम लोग पिलो कवर चेंज नहीं करेंगे ना तो मुंह भर पिंपल आएगा, और पसंद तो नहीं होगा तुम्हें भी पिंपल। और हाँ, पानी भी पीते रहना। अगर पिंपल पसंद नहीं है तो। ये तो भूत वाला गाना जैसा लग रहा है सुन के। अब मैं जाती हूँ, बाय एवरीवन, बाय बाय, गुड नाइट। वैसे मुझे भूत से डर नहीं लगता, मैं बिस्मिल्लाह कह देती हूँ ना। वैसे भी मैंने तुम्हारा शकल देखा है शुभम, मुझे तब तो डर नहीं लगा, फिर भूत से क्या ही डर लगेगा, है ना? ये रोएगा बाथरूम में जाके।